Hey everybody, good morning. You're watching the dive stream. We are live from RV Falcor 2 and you're watching a live camera feed from our subsea vehicle, ROV Sebastian. We really appreciate your patience as we've had to keep the audio muted for a little bit. We've had quite the morning of technical difficulties. This is dive 729. We are at a new site that we're calling El Quisco North. And due to technical issues, we're going to postpone mapping and we're going to do a little bit of sampling first. And with me here in the room today, I've got Doug and Tyler as our pilots right now. We've also got some folks working on the M3. I've got Juliana behind me. And she's also helping troubleshoot all of the issues along with our ROV team. So super busy morning we've had here. We've also got a room full of scientists. We've had quite the busy night of sample processing as well. And with me here in the watch lead seat, I've got none other than Dr. Lisa Levin. And so I'm going to pass over the mic to her, and she will be narrating this watch. All right, Lisa, over to you.
Good morning, everyone, or afternoon, or evening. As you heard, we're visiting a slightly different part of the El Quisco region right now. I missed it. Did we introduce our other people in the back yet?
In the back, no.
Okay, so we have with us, I'm going to let them introduce themselves. So I'm Lisa Levin from Scripps Institution of Oceanography at UC San Diego. Hachi? Oh, is she there? She's not on yet. No. Okay. Our loggers are Paula, go ahead.
Yeah, of course. Hi, everyone. I am Paola. I am this watch data logger along with Erin, who will be introducing herself very shortly. And I am from University of California studying microbes in the sediment. And
Hi, I'm Erin Fritz. I'm a data logger on this morning's dive as well. And I'm a microbiology student at Boston University.
Scott, why don't you introduce yourself to Hachi?
Hi all, I'm Scott Wyman from the Woods Hole Oceanographic Institution and MIT. I'm running the SAGE methane sensor on this dive.
Good morning. Mi nombre es Praxedes Muñoz de la Universidad Católica del Norte. Estamos prontos a tomar algunas muestras de esta zona para hacer algunos análisis geoquímicos por mi parte.
Okay, loggers, I hope you're recording tube worms, carbonates, and so on. So we came up here on the basis of some shipboard acoustic mapping, and as you heard, we were unable to do the ROV-based mapping, but we look like we've gotten pretty lucky in that we're headed to what's supposed to be a large depression with some reflectance indicating hard grounds, and looks like basically right at the start of the dive, we've hit tube worms, carbonates, and a few other types of seep organisms, and they're patchy and clustered, but it looks like a pretty active area. Scott, do you want to tell us, are we seeing any methane here?
Yeah, so we've been having elevated levels since we hit the seafloor, even while troubleshooting some other stuff. As we lifted off, it would drop back down to water column, and anytime we were within 4 or so meters of the seafloor, the methane values would start creeping back up again. So similar to our last site, even in these more background areas that we were in earlier, we're seeing elevated methane signals within the whole seepage zone.
It looks like we're coming up on a mound of some sort. Again, with lots of tube worms present. We have the tube worms in a couple of morphologies, the small clustered gnarly ones and the longer extended ones. Does anybody know what these fish are that are hanging out? You mean Maritza? Maybe she knows. That's beautiful. Maybe we could stop and take some photos here. Is that possible? Fish is pretty interesting, and I don't know if these are congria or some of the other commercially harvested fish. But it's kind of interesting how they've got their whole bodies sort of immersed in tubes. We also see a distinction between the fuzzy tube worms. Those fuzzy tube worms are covered with hydroids, which appear to be seep associated. Our shipboard experiments suggest that they may have the ability to use methane, perhaps through symbionts. But then we have these other clean, shorter tube worms that we're looking at right now. And I think we're waiting on Greg to sequence them and help figure out whether there are two species of tube worms here. Beautiful. Could you guys just mark BBC highlight, please? Let's zoom in. Can we?
Sure can.
They're so clean and uniform. And one has to wonder why there's no hydroids on that lower cluster. And there are hydroids all over all the other worms. Get a couple of good frame grabs. Yeah. So hydroids are cnidarians. They're colonial. So all that fuzzy stuff you see are a whole bunch of polyps living together in long branching strands. Traditionally, they're thought to be filter feeders or maybe even microcarnivores, but we have to wonder if they aren't here because of the methane.
I'll drop a marker here, Lisa. Do you have a name preference for this?
Well, we're at waypoint 1, so we'll just keep that name for the moment.
Okay.
And then we'll let people come up with something creative. So our mappers really did a fine job putting us on a great spot. So call out to Jeff and Adam. So the assemblage looks a lot like what we saw at the southern site yesterday. These coiled white snails, crabs, galatheid crabs, maybe a Munidopsis. But the fish are different.
They do appear to be different. Two different colors too, like one's orange, one's gray, blue.
Yeah, look, if we can get some video of the fish lying in the tubes, that would be
Sure, yeah, I'll see
really
if I can
cool.
do that. Let's get back on the St. Alex.
This is really an exploration dive in the sense that as far as we know, nobody's been here and looked at these features before. And any observations we can make about animal behavior and their distributions would be really useful. That is so amazing. I guess Eulohio left. I was hoping maybe he could tell me what these fish are. I suspect it's something you see in restaurants routinely. Oh, look,
Trying to
there's
frame
3 of
as
them.
I could in there.
That's a great picture. So the question is, what would they be doing there? Maybe.
There's a good little cluster over here too. Right there on the
Oh,
floor.
yeah.
One just went off frame. See it?
Yeah, there's another one under.
Yeah.
So many. So, I mean, if the fluids are sulfidic, they could be trying to get rid of their parasites. That's one possibility. They could be feeding, although they don't look like they're feeding. So, they could be hiding from fishermen. I don't know.
Don't seem to mind the crabs being right in their face though.
And maybe they're waiting for some cleaner creatures to come pick off their parasites. Hachi, you want to say something in Spanish about what we're looking
Just
at?
kind of observing this here.
Bueno, lo que estamos observando es una gran cantidad de peces,
There's more.
un tipo de congrio, no lo logró distinguir. Aparentemente es una zona de refugio, de alimentación. Lo que se está discutiendo en este minuto es saber cuál es la razón por la que se encuentran acá. No los habíamos visto en ninguna de las inmersiones del día de ayer, en zonas donde hubieran tubos de poliqueto.
So we're just observing.
We're trying to get some fish video. Oh, I feel like we should name this mound after the fish once we can figure out what they are. Maybe put out a message on the Falcor website for anybody with fish expertise. Or ask Eulogio to come in and help us. Looks like there's a plastic bag back there. I think we must have seen, how many fish in here? Probably 5 or 6. There's another one up. It almost looks like they've burrowed through the tubes and made little spaces for themselves. Some of them. See how many? Crazy. I mean, we have seen seep endemic fish hang out in tube worms, and there have been new species described, but I just have a feeling that these are not seep endemic fish. I don't think they're cusk eels, but they look a little bit eel-like. There's much smaller fish inside there. Looks like a Christmas tree with fish ornaments. Congrio. That was one of my guesses,
Yeah,
actually,
Congrio
about, I
colorado.
know so little about fish. Okay, Scott's showing me a picture that suggests maybe these are Congrio, and that's definitely something you get in a restaurant. I think I ate some a few nights ago before we left.
Por acá Eulogio nos dice que es congrio negro.
You think so? No. How wide would you say this mound feature is? Like, like a couple meters across
Yeah.
and high? Can you have any sense of how high it is?
La especie Genipteros maculatus, muy importante para la pesca local.
Vision.
I know it's a pretty decent amount, so I don't know what you want to call the bottom, I can measure it.
Thank you.
Just where the tube's at, or all the way when it's flat?
Well, the whole elevation of the feature, I feel like it's about 3 meters or something like
It
that.
might be more, let me see, so if I come down like that.
So now we're at 440.
Yeah, so let's see, how about,
So this is probably the base.
that's where we're gonna come up, let's see.
We're just trying to figure out how high this mountain feature is. Eulogio is writing, but it's Congrea negro, Genipterus maculatus. That's really important local fishery species that sells for $6 to $8 per kilogram.
So let's see, that is about
440, and we're at 434.
7 meters.
6 or 7 meters. So maybe one of the loggers put that. The height and the width is at least a few meters. I think we could, might be nice to do a suction sample here in amongst the tubes. And I've gotten back. Do you guys, I think we, I guess, have we been all the way around it now? Do you feel like you've
No,
done a full 3?
I'm coming that other way with, I get the
Okay.
last bit.
That little batch of cleanish tube worms with a different morphology is really interesting. They could be something different or homing in on a different set of conditions under there. Don't see any shimmering water or anything suggesting hydrothermal activity.
So that is, you've seen the whole thing now. And all the way around.
Sorry?
We made it all the way around.
This is the full circle. So actually, can you pan down just a tiny bit? I'm just curious about how many of those little clusters of clean worms, clean white worms we have. So it looks like 2.
Low, it might be some lower than that too.
Yep, there's another batch.
That actually looks like it continues all the way to the bottom of the mountain almost. As a patch.
Hmm.
Bubbles.
Yeah, so the big question is whether the more elongate worms covered with hydroids are the same or a different species than the small, tightly clustered and often curved.
You still see the bubbles?
No, they came right across the front of the screen. There were 3 streams, maybe, as we were backing up.
I might have disturbed him with the rest of that.
Yeah, could be.
Three what?
Little lengths of bubbles came by.
Bubbles. And you're seeing methane here, right?
Yeah.
A lot, or?
Good amount, similar to the smaller, the 2 mounds from the last site.
So it is actually pretty amazing that this mound is not covered with lost fishing gear, given where it is and all the fish. Has anybody seen fishing debris here yet? No? Do the fish, for those of you who've been watching this, do the fish seem like they're aggregated in one side of this? Mostly? Well, there's someone under. This is great viewing. These features are so big that they probably influence the environment in many different ways. I mean, clearly this is a place where methane's available to the animals, but it's the edifice is also going to alter the currents and probably how the methane moves around. Yeah, so this side has the most fish. Right here we can see 1, 2, 3, 4, 5, maybe more. These are good sized fish. I would say they're probably a meter long themselves, or 2/3 of a meter. And that direction was, well, we're headed.
Now we're facing west. Almost exactly.
That was west.
Yeah, we're
Where-
facing west right now.
It's the east, so that's the east face.
Yeah,
That's the big
yeah.
east face.
Yeah.
I see 4 or 5 fish here also. Yeah, look at them all down there. Lined up.
Lisa, the chat would like you to repeat what fish name you think it is.
We think it's Congrea negro, which is commercially harvested locally, served in restaurants throughout Chile.
See if you get that.
Thank you.
Super what?
And it's the observation of commercially important species utilizing seep habitats is really, really valuable information for us in terms of management for fisheries and management for conservation. It's not unusual to see commercially harvested fish at seeps in Southern California, where I also work. We have Dover sole and black cod and thorny heads. All are harvested commercially and all are really abundant around seeps. Probably they take advantage of the elevated productivity, but also the really strange reducing conditions that might be valuable for that don't bother the fish, things like hydrogen sulfide, but they do bother parasites and other unwanted creatures, so maybe the fish are hanging out in part for that and in part to feed. It's a very nice view of one. And you can see they're big fish. They look very eel-like. I guess they're sometimes called concreo eels,
It's like a cuskeel.
like a cusk eel. Yeah.
Yeah, they're also called a red cuskeel. Yeah.
Okay, let's stay away from the fish so we don't bother them and try suctioning in amongst the tubes just to see if we get a different community of associated organisms than we had a very similar mound yesterday to the south, about a kilometer to the south, but there were no fish inside, like no big fish like this. But this is something I've never seen any of the commercially harvested fish use tube-worm bushes like this in any other seeps. So that's a first. And it's wonderful to see it. The chat, somebody in the chat says they think they see about 50 fish.
Do they want to guess too? Yeah.
Sorry, say that.
Do they want to guess the tube worms as well?
How many tube worms? Okay. Yeah, we'll take tube worm counts here. I'm going to guess somewhere on the order of hundreds of thousands. So if you can just sort of push into the thick of them.
Anywhere particular, just to avoid the fish.
Yeah, just so we don't scare out the fish, but in the fuzzy ones, where the hydroids are.
And jar one is okay.
Oh, let me take a quick look. Jar one has a pretty fine mesh. Maybe use 3 or 4.
Index. Check the index on the pre-dive.
Yeah.
It's just. It's hard to guess right there. So devil's not reliable enough. Check altitude to see what happens. All right, let me see if I can turn the gains up. I guess. Remember what this would be on the floor. Starboard.
So in California and in Canada, I think Oregon and Washington, methane seeps are defined as essential fish habitat, which is a category that does allow some protection and suggests that they are important to the fishery industry. So this kind of observation of the cangrillo hanging out inside the tube worm mounds could contribute potentially to this kind of designation in Chile.
But it's on the
Perfect. And it's like that. Let me turn this back down before I forget.
So we're going to take a suction sample inside the tube worms, and that is to get some of the small associated invertebrate species that live in amongst and on the tubes. These tubes are really interesting in that many other seeps, there would be large numbers of snails and limpets and scale worm, polychaetes. We're not really seeing much of that. At least we did at the southern site. The hydroids seem to be dominant. Hydroids, as cnidarians, have stinging cells, so maybe they keep other things from settling also. The hydroids are the fuzz that you see covering the tubes. Try to go for the fuzzy areas, yeah. Because the white tubes look pretty clean, like not too much is there.
Just we're using such example.
3. It's flowing.
It would be okay to get a crab in there probably, although they're predators, so maybe we don't want them eating everything. What do you think?
Yeah, just they'll find something. Might have been posted.
Do you see
Something big
yeah,
going
sea
there?
star.
Great, it's fine.
And
Looked like a different sea star than what we had been seeing before. There's another small rat tail, like fish, also in there. So there's at least a couple of fish species in here. They're small lithodid crabs, these little spider crabs. We've also seen them on the bacterial mats eating bacteria.
Let's go to your right. You wanna try to get that fish? You, little one?
Do we want a fish? Yeah, I would say one of those. They look pretty small, don't they? But if you can't get it, it's okay. It's different, definitely. I wonder if the congrio are eating them. Those tiny ones.
They come in.
Yep,
Yeah.
got it.
Okay, let's go up right now. So that's
You got the fish?
Yeah. He's indeed.
You can see them in there. Yep.
That's what.
I'm just letting it float up so it's float.
So one of those fish is plenty. We don't, yeah.
No more fish.
Okay. Right.
Huh? Hey,
Go to your right. It's what you're in.
But we can keep sectioning because there's probably more things to get here. It's always hard to take these animals for science, you know, when they really deserve to stay down here.
move back down. Henry. That one's a little bit.
So what position?
Are these all tube worms? Tubes, that's not fishing line, right? That we're seeing in there.
Okay, this one. Right, go position.
Maybe one of the Galathea crabs would be good. That one right there, down there.
Here, see, Sunny?
Come down a little bit.
Yeah, you see which one she's wearing? Okay. That's it?
Yeah, great. Yeah, so the mollusks are rather few here, which is so interesting. Many seeps and vents, there are snails and limpets covering all the surfaces, tubes and rocks. So this is section 3, Pudge.
What are you trying to get that blue guy?
He wasn't any smart.
Ooh, could we, I know you're sectioning, but could we just zoom in on those two fish faces? Is that possible? Let's not section them, but
Would you mind
just-
running the camera?
just to get a really nice picture of them out there under the tube worms.
Okay.
It's a little awkward, but you know, it should be good.
Oh, maybe they don't like the lights. It's such a cool image. Yeah, okay, thank you. Got that.
So,
Yeah,
the
right
one
there,
that
right here.
opens it up, yeah.
Fish almost looks like a liparid, the little fish.
Yeah, if you drift into that a little bit, because it's gonna open up.
That's good right there.
Gotcha.
Such a nice view. There is no question the fish are using this space. But what they're doing here is a little unclear. Beautiful.
Oh.
These are the biggest fish I've ever seen using a reducing environment like vents or seeps, with the exception of some skates and rays that are also pretty big and come in, but
He's giving reverse.
No, but I can't. Okay, I can't turn on the floor. So when you do want to turn the light on?
Lisa, do you want the lasers on?
sure. Why don't we get a good idea? Why don't we put the lasers on just to get a size? I realize you've still got this section out, but
That's right. Let me turn it a little bit. So it's my heading up.
Excuse me. Yeah.
oh, look at all those fish there. Okay, so we can see the lasers. They're 10 centimeters apart. So this fish looks like a meter long. Easy. 10 centimeters wide. Did you guys get a couple of frame grabs with the lasers? Great. Okay. So they back in. They never sit with their heads forward and their tails out. It seems like they always see head out. So I don't know if they're watching for something. Prey. Fishermen. It's so fascinating how they're hanging out here. I think we can turn off the lasers. Yeah. This is probably a part of the fish life history that hasn't ever been described before. The use of this habitat. Don't know how, it's so, what's so fascinating is we were only a kilometer away at a big tube-borne mound and there were none. So maybe it's a local practice just for the congria that live here. Okay, let's go ahead and we still have space in section 3, right?
Yeah.
We can keep sectioning a little bit more.
Yeah.
And then I think we'll go ahead and leave this spot and go exploring to the next waypoint 6 after this. Yes, it's still section 3. It'll all be this from this big tube mound. So we got one of those pinkish starfish already.
Sorry about that. You want to stop it?
No, let's continue sectioning a little, just in case you can get some other invertebrates off the tubes or hydroids. I think we're good with the crabs, though. So just, we're after things you can't see that are microscopic. Is it a snail or?
Yeah.
Yeah, that would, could probably take one of them if you can get it off.
Yeah, something up there.
You know, put that in. Sure. If it's one.
So why was the African on the tonight?
You could, yeah.
Thanks.
Okay, got it.
Great.
And so for next
Snail.
one,
No, I didn't. What was that?
these two as well since this.
It was a white snail. Yeah. I don't know what snail that is. Greg says the snail is Calliastomidae. You got one right.
Yeah.
Great.
Okay, so maybe an extra line. Well, it's putting one right back where it has to be. Kind of showing up. Good.
I'm gonna step out. Things, but like, if it drops out, okay. But you're on iOS now.
Yes.
So you can keep on.
Okay. Sweet. Thanks, sir.
Okay, I think that's good. Got a nice representation of the different types of invertebrates and one small fish, I think, that are used in this habitat.
You're in the site there.
Yeah.
Oh, you should look at no juveniles.
Of the congria. There's a small species in here of a different fish, but yes, all the congria seem pretty well. So we're going to leave this amazingly beautiful mound of tube worms and investigate a series of reflectors to the west and see what else we find. There are still people that would like to have push cores. I would like to get some rock samples, so we'll go ahead and do some exploring and sampling. So waypoint 6 should be right there. We set up a series of waypoints to the north and to the, I guess, to the east now. And we have our choice, but the markers are closer to the east.
Bridge RV.
ROV bridge,
New waypoint.
copy that.
If we see any carbonate rocks that look small enough to sample, we'll stop and try to get them also. So these look like worm clusters. I don't know if they're on rocks. There's definitely a lot of tube worms here.
We have plenty of time to look because we'll be waiting for the bus. We have
Waiting
plenty of
for
time
the
to
ship
look.
to move.
So just let me know if you want to kind
Yeah. I mean, there is rock here.
Sixth grade.
of, yeah, but I'm looking at the trying to look, see if that rock will be.
This size rock is something we could pick up. But if the ship's moving, are we
We can
okay
stop
to
it.
work?
Yeah, we can do whatever we want, just as long as there's no restriction.
Okay. Well, there's a few rocks around here, so maybe we'd collect a couple.
Bridge RV.
Let them keep moving.
Yeah, go ahead.
Never mind, sorry.
So this way we can use that time while they're moving for a sample.
Is this going to be pickupable?
Yeah, I think so. There's one way to find out, I'm sure. Because that should be fine. They're only going to be how far from us. Yeah, that'll only be 135 meters. This could, okay, so you can measure.
So before we, let's zoom in on it, see what's there. Looks like there's shrimp, maybe a tube worm.
Would you mind moving it
That red coral is probably Swiftia. There's a crab,
Yeah, that's right.
sponge, some bryozoans.
Let's see how long it takes for them to do it.
The rocks often look kind of nondescript, but they're usually teeming with life. There's animals that live inside them as well as on them. There's some cryptic crabs here, maybe a polychaete. Not sure what that transparent, what that is. Let's go ahead and try to collect this. It's great. Actually, lasers would be good
Yeah,
if you
they're
zoom
on.
back out
Yeah,
with
just zoom
lasers
down
on.
too far. Or to it, far ahead. Let's see. Go. That's a little bit closer.
There we go. So rocks looks like it's about 15 centimeters.
Did we move our lowers up?
What?
We moved our lowers forward. Is there any shine the bright out of the?
No. I didn't.
No, I didn't think we did. So. Those.
We definitely can say that crabs like the seeps. Some people were hoping we would find Yeti crabs here, like we have at the Dancing Yetis in Costa Rica. Do you think it'll fit okay in the buy box?
It's close enough to try. We don't know for sure.
Hmm. What about turning and grabbing the top of it? Yeah. Curious fish. Okay. Then it broke. Okay, we might not take this one then, just because the creatures will have fallen off. Let's poke around.
Also, check the designations.
Yeah.
It's just some drawers and a bunch.
Do you have anything in the lab? Greg's in the lab. We'll look for a different see if we can find anything else that's a little more manageable. So we're now heading to waypoint 6, and there's definitely carbonates all around here. All of them are loose. Plastic bag of clamshell fragments. Anybody sees a live clam, let me know. A lot of crustaceans in the water. Presumably this is the migrating component that's down deep during engagement. And also we're asked to look for corals. These rocks here would be okay. This one has some creatures living on it.
Where?
Set up, you know, stop it.
On the seabed or floating? You see floating live or on the seabed?
On the seabed.
Okay. If it's floating, then I will blow up way, so.
Is this what you're, I think she's looking at all of
Yeah,
this.
I was just making sure.
This rock right here would be a great rock.
You're not cool.
Yeah, so,
First not went out.
Monster for sure.
Yeah.
Or this one, whatever is easier. We zoom in. So we can see some corals, some individual polyps, maybe hydroids, hydroids or anemones. The whole mesh-like pattern is bryozoans. Arborescent foraminifera. There's a crab under there. All camouflaged. Maybe tiny star orbs. The red is a coral. There's some little zoanthids on the rock, or at least tube openings. I'm not sure whose those are.
Maybe
octopus.
octopus.
I think that's an
A
octopus.
little white thing.
That's a cool.
I think it's a anemone coral. And these are bryozoans, these colonial
Sorry.
structures that are somewhat branched. They look a little bit like cactus, actually. It's great rock, got hydroids, lots of things. And a fish under it. Some crabs. The prelate amphipod is sitting on the red coral. That's that strange creature, looks like it's from outer space, that hangs on with its legs. Quite the garden. Sponge. So if you could get a good, frank grab of it, of the whole thing, that'd be great. Perfect. You guys got it? Sample. The fish is not gonna be happy if we take away their
The crab.
habitat.
It's churros.
Might be 2 rocks. Let's get one of them. Let's go down the shore. So it's pretty crumbly. Maybe, let's see, we don't want probably the one that's dropped in the mud. There's a way to pick up the other one piece. Might be useful. So all the bioboxes are open. Whatever's easy.
Did you want to try to take that piece above it, or just get it?
Well, let's just put it, I'm okay to put it in the box since we got such nice pictures on the ground.
Just that. So. Hold on.
Which box you got?
Maybe 4.
Go for it.
Fits. Turn it sideways. So 4B. Great.
So, yeah, plenty of room. So it's just head straight there.
To the next waypoint. Okay. That's good then.
What?
Bring it right on.
That's
What is it? I didn't see it. You can have the corals off the rock. I think I'm to tell me what we think. We'll see. But just don't hesitate to come up and say. So we're now heading to a waypoint to the southeast. It was also shown by ship mapping to be perhaps have hard ground. So there's solitary hydroid, the big one. There's rat tails, there's shrimp. Definitely mounds. Grenadiers, that's a different. Is that a cabria? Different.
Is there any particular you're looking at?
Well, we're looking for bacterial.
There's
mounds.
some-
There's something up ahead. Maybe go slow so we can take a look at what it is with bacteria.
Pretty much here, so.
We're at the waypoint. Okay.
Just gonna-
Let's just slow down and scan around.
just gonna photo this. Yep.
Marlo Lab, you want your samples in bacterial mats?
Yes, please.
Zoom in onto the seafloor.
Well, we're still quite far away. I'm just inching it slow so I don't kick up the sill. Get close. If when it's far away, we can't send the light that far. So we kind of have to get closer.
don't know yet. We'll see in a few minutes if it's here. It was much lower where we were sampling those rocks, Lisa. Also, was that a piece of wood behind the mats?
So it looks like plant. We'll take a look at it.
Yeah.
So, Erin, I'm gonna guess these mats aren't probably big enough for
Yeah, it's a little small.
what you guys want.
Yeah.
They're here. I'd like to zoom in and see if we see any animals specifically on them. Ecuador polychaetes or amphirettids.
I just was trying to let it go because I don't know if this is all that's coming up. Obviously, some of it doesn't even. You're right, look it up, kind of thing.
If the current direction's not great, we can keep moving.
Trying to figure out.
This is
Or is it just something all it is? Is it something photos?
Maybe.
I thought the current was going your head.
definitely got the plankton excited.
I look at that word for it. Just trying to step back to this.
So you guys noted that this spot, we're at waypoint 6, it has bacteria, small bacterial mats. Not too much else that we can see yet.
That's me. I hope this is-
Do we have any methane readings, Scott?
Not.
Yeah, that's what I was trying to.
I'm not seeing anything right now, but I would still give it another. We got here 4 minutes ago. Yeah, I would give it another 2 or 3 minutes before we know fully what it is here.
Okay, so while we're waiting, well, you know, honestly, I don't think it's such an important spot. We just want to take a quick look, but we can, we're at waypoint 6. We don't see any hard ground yet. Saw 2 little patches of filamentous bacterial mat.
So with respect to waypoint 6, which way would you like to, like from where we are now?
To the next waypoint. Waypoint 7,
Yeah.
but slowly, just in case we can see anything.
Bridge RV, new waypoint.
Open up.
I'm not surprised. I don't know, 'cause
I mean, are we continually releasing sediment as we go?
I don't know, I was wondering that, because I might have picked some up when I sat down with those rocks, but even when I come off the seabed, I'm not getting away from it, so it could have been stuff that I picked up on the way. Figure that out. So I don't do it again. Maybe since I'll be coming kind of close to the premise of that, like 55 maybe.
I mean, we definitely want to try to look at the bottom as we go, since there's nothing, but I don't know, visibility wise. So here is a row of burrows, a bunch of burrows. We don't know who makes them. I wonder if the shrimp burrow. I mean, we know Callian acid shrimp that live under the sediment will make burrows, but I wonder if any of the shrimp we're seeing here also dig burrows. I'm guessing we're in a euphosid patch, and these should be vertical migrators that have come down, and 400 to 450 is a good bottom depth for daytime bottom depth for these animals. Probably they're being attracted to our lights, but I'm not sure. It's certainly a productive area. Lots of food in the water. For somebody, I'm surprised we don't have any squid here. Yesterday we had squid, today we had congria. If we do see any plant or wood material or corals, we're after all of those things. So these are the big hydroids that we see. I can't hear. Sorry, but I think they're hydroids, but we haven't got a close look. So each of these waypoints showed up as a little white reflective spot on the mapping that was done. So the idea is there might be some hard ground associated with them. Or maybe it's under the sediment. Can we take a look at what those are? Could we zoom in on them? They look like, I know, some kind of cnidarian, maybe anthomastus, each with a crab and a fish. Interesting and pretty.
Staying on these or continue? Did you want to stay looking at these or continue?
We can continue. Did you guys get a picture? Yeah. So those are anthomastus, best as I can tell, which are like a pennatulid type of cnidarian related to sea anemones. Greg said heteropolypus was the other name for them. Or maybe it's a different genus, I think. That is interesting. Looks almost like a Larvasian house of some sort. Ooh, is that a coral? Yeah, it might be just following us and pushing everything in front of us. Well, while we're waiting for some visibility, Scott, you want to talk a little bit about what you're measuring and how? Is he there?
Yep, I'm here.
Yeah.
So yeah, we're measuring, actually, I guess now is a good time to chime in and say at those bacterial mats, Lisa, we did not see any increases in methane. And we're far enough along in time that we definitely would have seen anything associated. So we're measuring with the SAGE sensor from Woods Hole Oceanographic Institution the concentration of methane dissolved in the water column at these sites. While a lot of these fauna are associated with seep sites, we don't necessarily have good ways, or at least did not previously have good ways of directly confirming that methane in a real-time basis while exploring. So this is a in situ sensor that uses a gas permeable membrane to extract the dissolved gases in the water and then through an optical fiber measure the abundance of methane, and then as long as we know certain pressure parameters, we can calculate from that into a concentration of methane in the water. So we are measuring, as we're flying around, methane concentration. We saw many spikes over that large mound. It wasn't, super highly elevated everywhere. It was more heterogeneous than that, but we did see a number of big methane hits, and since we've moved on from there, haven't really seen anything. There was a low hit, what kind of where we were sampling, but that could also have been from not messing up the sediments, but lifting the sediments and releasing some of the gas that's under it.
Thank you.
Of course.
So I'll say a little bit about all these euphausids that we're seeing in the water. They are probably diurnal vertical migrators, part of what we call the mesopelagic community. We're pretty shallow, so they can't migrate any deeper than we are, which is 423 meters right now. But they're a really important part of the carbon pump. They feed at the surface and bring carbon back down to deeper waters. And that feeding on phytoplankton, which are extracting CO2 from the water, helps remove CO2 from the air. And the ocean is responsible for taking up a lot of the carbon dioxide out of the air and sequestering it down into deep water. And these animals that we're looking at swimming around here are a really important part of the what we call the biological pump, or the biological carbon pump. Most of the time we don't get to see this, the mesopelagic migrators in action. So it's kind of interesting. I think they're attracted to our lights, but they also live down at depth during the daytime to avoid predation in surface waters. These particular species also will have either had to travel through the oxygen minimum zone to get down here, or maybe in some cases they're resident down here. Okay. So can we just take a zoom in and see what that is? I don't think it's a coral. Some sort of Nidarian. Probably attached to something hard. It's okay, we can keep going.
In order to not pick up the seabed, I'm just obviously not have been using birds to go up, but even changing your heading, we're trying to stop. That's laterally, like 2 meters off the seabed, it'll just blow the birds. So I'm coming out of the fourth try. We're going to Yeah, everything's paving. I picked up a bunch of sediment, but I think I got most of it off when I set down the rock. And then any movement within 2 meters of the seabed, it flooded the big mass. And you've got a slight following current, so it's way through the whole way.
Right, Darby, did waypoint on light.
So I've been not using, not been pushing down in the stills. We're lucky.
Okay.
Yeah, so not, like, even with that intense.
So we're changing the pilot watch right now. Where did my
Yeah.
So when we get to waypoint 7, we'll just sort of look around for a moment to make sure, see if there's anything here. we're seeing lots of plankters. Some pretty close to the bottom. But nothing that looks like seep. Plenty of mounds and burrows made by somebody subsurface, some creature. Lots of shrimp. But we're basically at waypoint 7. Again, this was a reflective spot seen in mapping seafloor. No sign of tube worms, bacterial mat, clamshells. All our seep indicators, not even hydroids here. Nothing on the sonar, is there, that would give us a target right here?
Kind of decline up ahead, but nothing looks too hard on their turns.
Hermit crab with an anemone maybe on its back.
We suppose this area right here.
Oh, there's some rock. Okay, that's a good sign. We're over some rock. Carbonates. Okay, let's slow down and just take a peek. Coral, but I don't know if she wants this red one because I think it's one we've seen a lot of. But maybe we could home in on the rock. So there is, it looks like there's a bit of a carbonate pavement under us, right?
I'm gonna change my angle a little bit so I'm not blasting it.
So loggers, we can just say that waypoint 7 definitely has some carbonate, but wonder if there's anything else here. Doesn't look like it's particularly active with seepage based on who's on the rocks. I think we're at just a phase of the tide that is kicking everything up.
Yeah, it's not really clear and it just kind of hovering in this area, hanging around.
Is it ebb tide right now or something like that, flood tide? Or you think it's slack? And because I think the currents may be kicking up this sediment, not us.
Yeah, there's quite a bit of movement down there.
We see that sometimes in shallow areas. So let's poke around the rocks and see what's living on the ones down here if we can. What do we think the white is? Can't really see very well. Maybe serpulid polychaetes? Or something else. Can we zoom?
Yeah, I'm just gonna get a little bit closer.
Yeah. Greg said the polychaetes are unici. Inicid poly. So there's Galathea crabs. Maybe a couple of species there. What? Do we want it? How do we know if Anna wants this coral? Anna, if you're watching, can you tell us if you want us to take this rock for this coral? Substrate that shows up out here. So some are under it, some are in it, some are on top of it, some are attached to it.
Oh, yeah.
What happened?
There's a lot of shrimp in the filter.
Oh, yeah. Well, we could try collecting this rock and see what we get. There's a variety of things on here.
They have to put it around for you. They may have to put it
Can
around
we-
for you.
A black coral?
Oh,
Okay, wait, hang on. She's looking, she wants to look at this coral. We're trying to figure out which coral she needs, so. Well, no, I'll collect the other rock if you want that coral. I don't care.
If you collect the other rock, it would be better. If you would like.
Can we take a look, sorry, now that you're zoomed in here at that
Big
rock?
one.
Yeah, up there. Just don't go away, stay here and just,
I'm checking because
is this a coral you want?
yes, I want that.
This one.
Yeah, you are collecting.
Do you want the whole coral? Collect the rock if you want the whole coral. Or do you want to just cut a piece of it?
Just a piece.
Just a piece.
Unless you need the rock. Up to you. Uy, nice.
Do you think that rock would fit in the bio box?
It's quite large.
Yeah, it's a big one.
Too big, okay. Then
So
let's
just that
let's
piece.
go ahead and get a piece of the coral.
Okay.
And I'll worry about the other rock later.
Just to go in a quiver.
In the coral clipper, yeah.
How close on that, Doug?
We'll see.
Yeah. Just a little bit closer.
Kick it back out. Okay, now bring it in.
Está intentando tomar una roca que tiene varios organismos, incluidos corales y aguafilas, los cuales han sido muy devastados con la pesca de arrastre, que es muy importante conocer cuáles son todas las especies de corales
Good.
en este lugar.
Yep. Oh, yeah, just a piece of it, right? We're trying to cut it. Okay. So that rock's interesting. There are about 3 different kinds of crabs hanging out underneath it. Some pretty well camouflaged. So, is Anna still here? What coral is it? Can you tell our listeners? No. It's an octocoral.
Yeah, but I don't know if it's.
We don't know what- It's a gorgia, maybe a lepto or a paragorgia. Our coral expert is telling us. It's a nice little amphipod on it, and I'm not sure what else we see there. And it's going to go in Coral Quiver 1. So we're at waypoint 7 right now. Sticky. Yep, there it goes. Thank you. So we use these quivers for any biology, but they've been given the name coral quiver, so quiver. All right, we might go back and try to collect that other rock we were looking at, right down here. This would be classified as an inactive seep area or transition, maybe. We don't know that there's no seepage here. There had to have been at one time because we're sitting on a carbonate pavement. And these are orthogenic carbonates made by the microbial consortia called anaerobic methane oxidizers. They consist of bacteria and archaea living together. The archaea consume methane and provide organic matter to the bacteria, which then produce sulfate and produce hydrogen sulfide. And a byproduct of all of this is the production of the carbonate rocks that we're looking at here.
This one in the middle right here?
Yeah, it's great. If it's small enough. Sometimes there's an underground component that's and also make sure something else bigger.
Lisa, for what it's worth, I haven't seen any methane bugs seafloor background
Right.
in this area. I've been around here for a while, so
Yep.
I'm starting to see that.
So I'm pretty sure this would qualify as inactive area. And it's sort of interesting to see what
That's a bit deceiving.
lives where the rocks are in seepage versus where they're not. Is that too big?
Yeah, it's attached to the one behind it and
Okay.
see how everything.
Okay,
moves.
that's fine. We'll skip it.
All right.
That's why I said
Yeah.
if it's small,
It's
but
deceiving.
you can't always tell
Yeah.
surface manifestation. All right, well, let's just, so I'm gonna say that this is not an active area, but I wonder if we should just wander.
I put in, this
Yeah.
is the more cleaned up version, and we're right here, and this was the first place you were sampling.
Yeah,
It looks like there's some pretty high mounds and large features near the end of the waypoints.
so maybe
But
we should.
not-
As far as the end of the waypoint, we could go over there, just passing through our waypoints enough to see. And which waypoint number is it, Jeff?
I need to double check.
Like 12 or 14?
So to get to the highest mound in front of us is 300 meters. And
It's not so far, actually, but which of these is it, do you think?
So we're here.
it's gotta be one of these, 10 or 11. It might be 11, but this map is not as shaded to easily see it, but I think these are gonna be mostly inactive. If
Okay.
you found that one at, are you right now?
We're at 7.
Yeah, because they look about the same, from 7 to 10. It's like the same type of feature. So maybe a quick pass over,
You will
but
just,
you can get.
Yeah. So much better on the light.
Red ROV, got an updated waypoint.
so we just had Jeff Beeson come in and tell us that they've cleaned up the bathymetry and they see a larger mound further down our waypoint train. So we're going to move quickly through the next few waypoints, try to find another large mound of potentially of tube worms.
So, into the depression and the mound. And the shipboard mapping, it's a broad hole in the middle of this mound, which was the tube-worm mass.
So it's about 300 meters away, but we aren't taking the most direct track because of our waypoints. Ooh, there was something off to the left. Can you slow down and veer left? Oh, yeah. Bacterial nets. It's really easy to go by things and miss them because everything's patchy and there's not much light out here. Okay, Matt, people, like Aaron, what do you think?
These seem pretty big. These seem like a good size for the course we made. It looks like we could possibly get the fibrilla ones, maybe in
Great.
this
We're gonna
cluster,
stop
and
here.
then if other people want see that course from the same area, it could maybe go up. So.
Okay. So I think we're going to try coring here.
Scott, do you have a? You see me?
This is more or less the biggest bacterial patch we've seen today.
If only it was that quick.
Sorry.
I will know, Aaron, by the end of coring, I'll definitely have enough readings to say whether it's different. Given what we saw yesterday and the day before, I don't expect signals here.
Okay.
Yeah. But I can always be wrong.
Not wrong. You're just not right.
So we are, interestingly enough, we're not at any waypoint. We're between 7 and 8. But it's quite likely that there's a string of something subsurface because all these waypoints are more or less in a line. There are dead clamshells here as well, but no sign of living clams.
I can do a little bit more on the starboard arm.
Before we take any cores, maybe we can zoom in and get just some photographs of the map itself. There was an acarax ahead. So acarax is a type of clam, a solomyid clam that has symbionts. It's that thing in the distance that you see with a little dark, it's very dark. The periostracum is an interesting and characteristic shape. So, but acarax has sulfide oxidizing symbionts that provide its nutrition. But we're going to take a series of push cores here for the Marlowe microbiology team and for Pachi. And we'll see if we can fit a few others in. But so let's just zoom in and look at the surface, see if we see any of our typical friends, amphoretid polychaete tentacles or dorvaleids. Okay, we definitely have amphorettids. If you guys with the sharp eyes can see the little pairs of red tentacles sticking out of the mat, those would be amphoretid polychaetes. We don't see any scale worms or dorvaleids crawling across the surface. These bacterial mats are mostly made of Beggiatoa-like microbes. Under a microscope, they look kind of like spaghetti. And they tell us that there's quite a lot of hydrogen sulfide underneath. Okay. So Erin, point again to me which part of the mat you want sampled.
Just for the sake of getting 5 nearby, assuming,
You want the widest patch?
yeah, the wide patch, yeah.
So she wants, we need 5 cores, sort of side by side in that middle. I think the middle part would be the widest area.
Just doing 5 here?
No, we're gonna do a couple others.
Okay.
But those 5 need to be together, and then we need probably two more.
Okay.
2 to 3 more, depending on what we can fit in. If we can get all the mat coring done here without having to stop again, that would be good. So there's 1 for Pachi, 1 for potentially Victor Gallardo, and for maybe Jorge. There might even be 1 more. And Gracie wants 1 also. So we're up to 8. 8 cores, but the first 5 need to be together, and then the rest can be in different parts of the map. And if you don't mind putting them all in before we pull any out. That would be great.
All 8 or the 5?
8.
All 8
8,
in.
but 5, yeah, I'll 8 in before we remove any. However, if there's a carbonate base down there that we can't get through, we might not want them. So Aaron,
That's as deep as
let's
it's gonna
decide.
get. But,
Is that gonna be deep enough for you?
That's probably about 6 centimeters, maybe.
Should
Yeah,
we-
I mean it's. Not sure.
Do you want us to try another one? See if it goes in further?
Yeah, if you could just try another and then decide.
Oh, it seems like it went in a little further.
That's it for that one. That's as deep as that one's gonna go.
What do you think?
About the same as the other one.
I mean, this is the biggest match we've seen, right? I think I'd rather get like really nice matting than somewhere with less mats, but deeper. So I think, Hachi, would you still want to go over here, or would you want?
I would prefer
Okay.
Ooh, there's a liparrid fish. So we're having a discussion among the core users about whether these are going to be deep enough for their needs. Hang on, we'll decide in a second.
Already big and deep.
Okay, we'll just proceed, and maybe we can find it, one of them can be a little deeper.
Yeah.
You want to keep pushing cores in here?
Right, we continue?
Yeah, yes.
Aparentemente no podemos tener testigos más largos debido a carbonatos que están bajo la superficie o también agregaciones de carbonatos biogénicos, los bivalvos que se desarrollan en esta zona. Observando los flujos de metano, los sensores no son tan altos, pero esta es una zona bastante grande, lo cual la sumatoria total de la cantidad de metano que se genera en esta zona podría ser bastante grande, bastante difusa. Y por eso las agregaciones de parches de las papeles bacterianas.
This is okay here?
I think they want them all in that
If you could
big
do it kind of
patch.
behind 6 or 5.
We're gonna have to move that.
Oh,
Not gonna reach there.
How about in front of 6?
There's no room there.
There's no room.
Behind, could you reach behind 7?
If we move, yeah. We can move forward.
All right. Coming up. Up there for you.
I doubt it. We'll try. One more here.
Yes, please.
Hachi, can yours be in any part of this mat?
Close to them.
Close to them? Yeah, okay. Almost looks like there's a clam in that one.
That one's not going at all. That one didn't go down very far at all.
If you wouldn't mind shaking it out away from the seat, and then maybe the application.
But there's three, it looks like 3 clams in there, doesn't it?
I'll just see one of these then.
I don't know. Just shake it out, it's fine.
This is okay to shake it out here?
Yeah. Further off the seat, can you swing out a little bit further away? Yeah, so that we
That's over
get
the
some.
top of that.
Because it's not coming out.
Can I see, is it still, if it's still in there, we'll just take the core.
It's not,
It's gone?
yeah.
Okay.
It's just dust.
Okay.
Is it cool? Okay, perfect.
And we want 3 more in bacteria somewhere near these. Pachi wants one near these for sure. I don't think the other 2 have to be very close. So, yes, hold on 1 sec, please. This is your core. Can you direct him to the right spot? And then, yeah. Are you okay with this? Pachi? Yeah. Closer to what mat? I mean, if he goes down into the white mat there, is that okay? Yeah. So just this side of 7? Yeah? Yeah, great. Okay, and then 2 more, please. I know, but 3 other people do, so. Gracie wants one, Gallardo wants one, Jose maybe.
Still got that condition on it.
That's Gracie, yeah.
Yeah.
Maybe we'll even take 1 extra because something's gonna fail.
Where's this one?
Is this a good spot right here for this next one?
Sure. Davide needs core. And Victor Gallardo needs a core. Pachi needs a core. And one of these is sure to fail, so we should take an extra one, right? So maybe one of the microbiologists in here can tell us why microbes are so interesting at methane seeps. What's special about these bacterial mats?
It's a good spot here too.
Yeah, perfect. It is. I can hear you.
Yeah, it's a great question. So, as you can see, just as how we have it in the
send out lots of methane or increases gas, the lowering potential of that.
It's heating.
I'm carbon dioxide in the next 100 years or so. But upwards of 80 to 90% or so.
I'm gonna ask for one extra, just in case
by
we
the
lose
microbes
one
in
of
the
these.
atmosphere. That's mostly attributed to archaea known as anaerobic spectrums or ANB.
Because they're all spoken
They're
for,
capable
but
of
somebody will be
oxidizing
unhappy
methane
with
and
all
in a
that.
partnership with sulfate-producing bacteria. So they're really powerful little carbon sinks in there. Another really neat thing is that they produce carbonate rocks, which is the reason the geomicrobiologists from Earth are so interested in these structures as well. Because the geographical environment in these has changed so rapidly to get some very interesting illustrations in just even a few centimeters of sediment. So when you treat these shores, you be sure to switch them up horizontally into little pots almost. And you can see a lot of variation and diversity in the microbes and what they're doing, even just within the centimeters of these relatively shallow voids.
Okay, get a frame graph, please, of all the core is laid out. You guys good, Logger? You got a picture? Yep. Great. So we're at 427 meters at a white bacterial mat that's about halfway between waypoint 7, a third of the way towards waypoint 8, after waypoint 7.
Are the 5 in the middle the priority?
Yeah, we'll go ahead and take those first 5 first.
Just 5 first?
Well, whatever, actually, it doesn't really matter what order you recover them in, whatever is easiest for you guys.
Full of bubbles.
So, whoop, bubbles. Methane's here. You can note 7 has bubbles. Yep.
One bubble at 5, so not many.
Nice. Good. Okay, this one.
If you could just shake it out instead of putting it in so we can reuse that one.
Do you want to try punching it again?
Yeah, that'd be great, thank you.
I can't reach anywhere else.
What about next to 10? Would that work for you guys or not?
Yeah, that would be fine. We,
Just on a, a white spot. Yeah.
That's not good. It's too shallow there. That was awful. It's too shallow.
after we pull 10 through 13, maybe could we go back here and try again with 8 and 9? Possibly.
So do you want us to put that in now? Or do you
Um,
want to pull the other ones out first?
If you could just put that one aside and pull out the other ones, so, and then possibly sample up here with 8 and 9,
Yes, of course.
if that is possible. Just so that you can move without those being in the way.
So they're all pretty not sticky.
Shallow.
And short.
Yeah.
That's good. There.
Mm. Looks good.
Have we assigned core 11 to a group?
Who are you assigning it to?
I just mentioned that Opal still needed one in Vector.
I think the thing to do, well, I guess you can tell from here whether they're gonna be good enough. Hachi should have her choice of which of these she wants, I think. She can see them all. That one's seeing a little bit of the bottom. Are you still happy with number 10, Hachi, or?
I would prefer 11, but.
Yeah, so I'm happy for you to have 11.
Well, then what's so good going on? Just,
It's always a challenge to take good deep seep cores because some of the properties that make them good, like anaerobic methane oxidation, also lead to properties that make them fall out of the push cores. Little bits of carbonate, sometimes bubbles. All of these things can disrupt the cores. Okay, so Erin, you don't want to use any of these ones we just took, except the first 3, right?
Yeah.
And
If
then,
you could try again with the 8 and 9 in this area, or above where the others were. Otherwise, I could use,
well, should we collect 9 and see how it is or not?
okay.
You willing to use it if it's,
Yeah.
if it stays in?
Yep.
Okay.
That's gonna fall right out, it's already loose.
Doesn't look like it's gonna stay in, does it? No. Okay, so let's shake that out and we can try it again, right?
Yeah.
So you're looking at, just to be clear, this area center screen, with the lasers around.
Erin, is this what you want back there?
Right here.
Alright,
She's
so
pointing to this, yeah.
Yeah, sorry, I can't see your screen from here. This one.
Up.
After we get these 2 cores, I have a sediment scoop requested, so.
I'll try and go. We want this right here. Right here. This is this spot, right?
Yep.
Okay, alright. That is very shallow. That's not it.
Sorry to be a pest, but I might actually ask that this next one goes above it so that the sediment is not covering the mat. Yeah.
Out there on that white patch.
Yes,
Okay.
I'm sorry, could you also possibly put 9 next to it as well? Just so that I can explain, but if you're interested, but it would be wonderful.
So you want to shake out 9?
Yeah, yes. Really like water ride.
There's some bubbles at the top of it. There we go. There we go.
That was fun. That's how I was in the room when they push course. Attention.
Ooh, bubble stream. This has been a hard set of course to map, for sure. I keep drawing them and then crossing them out and drawing them.
Diagram,
Okay, I actually think there are some living clams here, and we'll try to get them with the sediment scoop after we're done coring.
You good to grab these? Y'all want to park them a little bit longer? Good?
If waiting longer would help, then we can wait a second. I don't- Is there an advantage to waiting?
No, I would collect your cores,
Alright, sorry.
so they don't get knocked over or whatever. Bubbles.
Yeah, that's all right. The bubbles are what's making the core that. As soon as it pops the bubbles up. Yeah. Nice. This is weird.
Live plants?
Thank you so much.
Super, thank you.
That one looked pretty good. Alright. Just keep it out there. I think she said we're gonna do a scoop next, so I'll have to grab one of those, okay? Is that correct? Lisa?
Yes.
Okay.
Try to scoop.
Just make sure your camera's back. Can't get at you.
It's right here.
Everything is in order?
Oh,
Oh,
Iskin.
Yeah.
You know what,
Thank you.
before you do that, is it possible that, sorry, we forgot, we should have done it
Yeah.
first. The scoop will make it worse, so we should,
I won't scoop, I'll just get it ready and then
we'll take a
do the
Niskin.
Niskin, no problem.
Yeah. Is one Niskin gonna be enough here, or do we need two?
Okay.
One will be enough for me. And I can also, I won't need to hold this in, so I can share it with somebody. So.
You probably should have taken a Niskin over the big tuber mat. Next to our mount will do it.
I just don't have it on. Sure. Sorry about that, Joe. All right, Tyler. Try to lower it some. Open it up here. See where we changed it. All right, let's change that valve real quick. Ish. Okay. And retract. There it is, this is one.
Okay, this one. Over the bacterial mat. So for scooping, I think if you can scoop some of the mat and some next to the mat of the sediment that's not
Any
late,
preference?
as to where?
Yes.
Maybe near the clams,
On the
somewhere in,
undisturbed side,
if,
over
can
there?
you reach
Okay.
this? Whatever you can
I-
reach is
I think I
okay.
can probably reach that.
Probably not here, where we dug up the bottom. We just really probably want the top 4 or 5 centimeters.
Of course, that, I'm not that way.
I do think I saw a couple of potentially living clams, so hopefully we
Did ya?
could get them in here.
Where do you think you saw those?
Oh, in the push course.
All right. Well,
But
let's
it
be
just,
on
just, this is great. We also, there's potentially an acrax on the other side that we might want to try to pick up. But let's start here.
That's a clam bank.
Does anything look alive in there? You can't really tell, but
Couldn't tell.
that might be, yeah.
I was out looking for you.
Maybe another scoop like that. Some not in the mat also, some just in the mud next to it, yeah.
I don't know if I
This is for to get animals, so. But we want to try for the amphoretids and Olamyid clam. I don't think it's alive. Usually when they're up on the surface like that, that's a dead periostracum. They live under.
What's the request?
The request is just to look at it
Oh, sure, yeah.
while you're waiting for things to clear.
Oh, you
And
wanna-
and then,
Sure,
I don't think
sure.
we can collect it,
Smash all the way.
Not from here, we can move and collect it, but.
yeah. But it's not alive, so I'm not sure collecting is so helpful. But it is the first one we've seen on this
Oh,
entire
okay.
expedition.
All right, cool.
So it's exciting because it
Like
is
your
a
stuff.
sea clam.
Your stuff looks good.
Yeah.
Alright. Maximum.
Okay. So it's fine to just do another scoop that's sort of next to the bacteria mat from the Yeah.
Should I label these as sediment scoop 1 or 2 within the bio box?
I can't tell, but do you know what number scoop that is?
2.
2.
Thank
Yeah,
you.
two right there on the handle. Stuff is so
It looks like jelly down there.
Marshmallows. I love the marshmallows.
Deep sea s'more.
High water content.
And then you
Great.
Got some mat there. Yeah, got some mat.
Okay, that's great.
You want some more? Are you happy with that?
That, I'm happy with that.
All right, which bio box would we put in this bad boy?
Whatever is handy, anything but 4B.
Anything but 4B, okay. Sounds like a plan. Let's go for 2. Or 1, whichever one.
Alright. Sorry, I'm not giving you any.
That's okay, yeah.
Okay, we're taking 1B or 1A?
I'm gonna try for 1A, but
Great, thank you. Okay,
anything else here?
we got our Niskin, we got our sediment scoop. Anybody want anything else? Hachi, do you need a Niskin, separate Niskin? So you share with them? Anybody else? Okay, I guess not. Let's head to the next waypoint.
You got 8 there total, right? On the push course.
I'm sorry?
We did 9. So 5, 6, 7, 8, 10, 11, 12, 13.
Third.
Alright.
So you guys wanted one more this week?
No.
No,
I
okay.
think we're okay with the Niskin. I probably should have taken one of that giant tube around, but didn't remember.
Bio box humor today.
As per request.
Is it 2B or not 2B?
Finally, somebody noticed that.
Alright. No more sampling?
That's the one guy with a master's in classic literature.
That was, I take no credit for that.
Okay, no more sampling here.
Alright.
We will head to waypoint, we'll continue towards 8.
Let's do it.
We even left a few bacterial mats on the, unmolested.
No evidence we were here.
I can even say that.
All right, where are we headed? Back on our route?
Yes.
Okay.
I guess we went off track by a few meters to get to this map.
Right, are we?
It's a nice aerial view of them.
Yeah, go ahead.
Yeah, Tony, we can continue to that waypoint. Thank you.
Copy that.
So we're heading along a set of waypoints that ultimately we think lead to a fairly large mound, but the smaller waypoints show some reflectance in the mapping that should represent carbonate, authigenic carbonate rocks. So each waypoint, we just will scan around the bottom, see if we see anything. It's sort of interesting how abrupt the transition from seepage to non-seepage can be on the seafloor. You'll see little patches of bacterial mats like we were at, and then just a few meters away, there's no evidence of seepage. I think because they're so small and patchy in many cases, it means that the entire continental margin is really affected by seeping, and the animals here move in and out of the seep at will, probably transferring energy, carbon, methane. Methane moves through the water, so any of the animals that have symbionts or feed on bacteria that use methane can access it through the water as well as below ground. Scott, you never saw any elevation there, did you? Or when the bubbles came out?
When we were pulling push cores out and sediment scooping, I'm seeing some peaks essentially now from that.
Okay. So these are more of these cnidarians, heteropolypus. I think Anthomastus is an old name. Oh, we're coming up to some tube worms. So waypoint 8 looks like a real thing. Let's take a look here. That's pretty neat.
Some mats.
A small mound. We can see it slightly elevated with tube worms and some white bacterial mats and some carbonate. With the trusty corals next door. Increasingly, we don't think of corals as being seep organisms, but the red one, which I think is Swiftia, is very, very commonly found around seeps. We saw that at Costa Rica as well.
Explore this a little bit.
Just scan it around to see what we see. Zooming in a little bit in case we get any new creatures. If we see any rocks that look like they'd fit in the bio box and could be picked up, I would like to have one, but I don't see any here. Oh, fishing net. Yeah, you guys can record that. Probably a bottom trawl. Yeah. I guess we could pan down and photograph it. It's pretty. The thing about methane seeps is they are on continental margins close to shore where human beings have a lot of activities, including fishing. So it's not that uncommon to see fishing gear, fishing trawl marks or even fish tangled up in nets. Little nets like this on the bottom, we call them ghost nets. Sometimes they continue to catch things, but of course they'll never be recovered.
How long do you think they have to be at the bottom to grow that level of biofouling, Lisa?
Oh, a while, but I don't know how long. Maybe just a year or two.
Yeah.
I once had a set of experiments put out at 500 meters off Central California, and I came back and clearly my colonization trays had been tipped over by a trawl net and were upside down, along with some trawl marks. It's one of the hazards of working at seeps because they're so close to shore. We see discolored, sort of gray-brown, grayish sediment here, in contrast to the normal brown color. So that indicates they're reducing with hydrogen sulfide present. There's a hermit crab. Ooh, is that wood? Could we just take a gander here? See if that is a piece of wood, or if it is, we'd like to collect it.
Yeah, it looks on the left side there, there's definitely shape.
Does it look like something man-made or just?
It certainly does, that looks like wood.
It looks like
that.
Yeah, nice.
You know, lobster pods.
Yeah, that looks like
We're interested in collecting wood on the seafloor. But let's just take a look at it and see what we see on it first. And if it's a size we could collect even. Greg Rouse studies wood fauna. I sometimes do wood colonization experiments. This, let's see if Greg's able to tell us if he wants this. Before we collect anything, we'll just
Yes.
It looked like fabric.
Oh, it does.
so maybe it's not wood?
Do we want
No,
fabric?
that's definitely like fabric.
Fabric
Yeah, I think it's good.
disguised as wood?
Yeah, it looked like it had,
It's been honing, it's camouflage.
Okay, not wood. That's weird. Do you think that whole thing here is?
It'll fold it in the box, right in there. Let's see what's looking over here. Yeah, that's definitely like fabric.
Hmm.
Yeah.
Huh. Let me see.
Yeah.
Oh, we can
I'm
clean
not gonna pull it all the way free for fear that it'll end up in my thruster, but I just wanted to give you a good look. It looks like some sort of maybe netting. Is that like maybe some wire
Yeah,
around the outside?
it's almost like basket,
Yeah, something.
where I'm like onion, what do you call those?
Yeah, data sack, I think
Sack,
type thing.
yeah,
Burlap. Yeah.
burlap.
Burlap.
It's hard to find a seep without human debris here.
Right?
Yeah.
Okay.
So there's, how about methane, Scott? Are we seeing any?
I think we're still coming down a bit from those bumps in the sampling. We'll, in another minute or two, should start to see if there's a signal here.
Okay.
Do you want me to hold still, or is that?
No, I think we can probably continue.
Yeah, I would agree with Lisa.
to the next waypoint.
Okay.
We know that there's definitely a seep here, seep activity.
Detritus.
Is that like a trawl mark? What are we looking at? Does that look like something was dragged through the mud
Sure
there?
does.
Could they be troll marks?
Yeah, could be.
This wood right here?
No, it's a plastic bottle.
The bottle.
Green
Yeah.
plastic bottle.
Sprite.
Alright, I'll get back on the line.
And I think that I'm working down my waypoint 9.
Do you waypoint?
All right, Darrell.
Yeah, we are moving now.
Thanks, Tony.
This is, as far as I know, the first time anybody's really had a good look at this part of the seafloor. I think nobody knew El Quisco would have so much, well, such large features like these giant tube-worm mounds, and so many patches. We found quite a few patches on the seafloor thanks to our geology mappers, Jeff and Adam. It's interesting, we haven't seen any of those cangrejo anywhere else except hanging out in the tube-worm bushes. Anemone. You're free. See something on the sonar?
Yeah. See here, these kind of drag marks? And then you have this little.
So
indent
trawl,
here.
bottom
Yeah.
trawls.
Yeah, this is probably waypoint 9 right here.
Want to sit down and observe the glory of this coral for a little bit? Go to the mound or?
Yeah, we've sort of arrived at a seep site. I'm gonna find out if our coral person wants a piece of this. Looks like an octocoral with brittle stars. In the back, a whole bunch of shell hash and some tube worms.
Good, Garble, you were gonna stop and investigate here for a little bit.
Okay, copy. Stopping here.
Next one.
So yes, she'd like this coral snip from this into a coral quiver, please.
Nothing shifts around too, but it looks like
It's a primnoid.
Yeah.
So there's a small carbonate mound here. We're at waypoint 9. We're just about. A lot of dead vesicomyid clamshells. Vesicomyids have symbionts that typically oxidize sulfide. So they're a characteristic seep clam. Whether the corals are here because of seep productivity or because the mound enhances the currents and helps them feed, we don't really know why these corals are sitting next to the seep.
That's all right, I got you.
It's really nice to have our coral quivers attached today instead of lying on the bottom somewhere.
They're more useful this way.
Very helpful to have them. Okay. So that's number 5. They're opening. Yeah, thank you.
Any particular branch, dealer's choice?
No, I don't think she cares, but probably if you can avoid disturbing the brittle stars, that would be good, but maybe something from the top or. I don't know what that is on over there.
What's that?
Looks like a kiwi sitting on the.
Oh.
Oh, it's an egg case. It's a shark egg case. How nice.
amazing.
We'll have to take a picture of that eventually.
Oh, nice.
I think we don't want to take the egg case because that'll be some skate or rays that will not hatch. Corals are often used as egg case sites. I think it's an egg case. It's a little hard to tell, but. Very nice, thanks.
Yeah, that's what I was going for. I thought I was caught up with everything else.
So it's a primnoid. Don't know which one.
Oh, did you need a close-up shot? I'm sorry. Do you want a close-up shot before I put it in there?
I think you can put it in, but we might go zoom in on the
Okay.
remaining coral and see if we can get pictures of the tentacles. Great, thank you. Really nice. Let's get a shot of the egg case. That's what it is. It's one of the things about the deep ocean is so many animals use the large structure forming.
Oh, okay.
Yeah. Did you guys get a picture?
Yes.
Okay, great. So can we zoom in any more on the polyps? Or is this max?
It's max, but if you minimize it's close to one.
Okay, just so we have a picture of the extended tentacles and so on. I know the scientists count them and beautiful. Lovely.
A little closer for me as well.
We can get closer.
Yeah, you can smooth this stuff up a little bit.
Okay.
May kick up some dust though, so that's the.
Well, I'm gonna guess this is fine
Okay.
for, well, it says if possible, a good zoom in the polyps, so I guess they want us to go closer.
Okay.
We'll see how. I don't know if anybody notices these little transparent creatures floating by their Keating nests. I think arrowworm is the common name. I rarely see them. They're showing up really well here.
Yeah, I got a little bit more actually. Breakfast whiskey.
So we've been asked for some super zoom of the tentacles. That's pretty neat. Beautiful. Push a little BBC button there. Yep. These orange brittle stars live as commensals on the coral. And you see they're up there for feeding. I'm not sure if they're feeding directly on the corals or just filter feeding. The polyps show up pretty well as well as the tentacles, so I think we can probably count 8 tentacles, making them up to corals.
It's a little silty. It's been worse.
For a shift change. We will be very soon.
Alright, who wants to fly? Awesome. We are following the waypoint. We just have to look at the squirrel. If there's anything else in this vicinity they want to look at.
These three, these are maybe a little bit more than that. These two are
Projects. So we come out and just.
Just wanted to let you know our navigation has been solid for the last few hours, so if you guys did want a map, we can. Just so you know.
You can do that. I've been done it, so I haven't set it.
So, I'm sure we're just going waypoint to waypoint, but there should be a giant mound over, it's either in this, it's either 13, 11, 13, or somewhere in there.
But some of it's.
Sort of like what we were at this morning, probably, or bigger.
It's a little dynamic.
But I figured it was worth just looking at each waypoint, and sure enough, there's seep, something seepy at each place. We are at 9. Yeah, so here, it won't be too far to go. Should be fun. We got 1 rock so far. I think most of the push cord needs are met now, so
Yeah.
shouldn't
Yeah.
have to do much with that. Like you? Yeah. And there's lots of empty spaces for them. So, I mean, I don't think, if you saw great things like clams, you could push for them. I couldn't tell if there were any live clams. There were clams, lots of dead clam shells. I thought I was seeing live clams go in the push cords that were taken for the microbiologists, but I'm not sure about that. Not most, but, and maybe one that fell out. There should be 1 scoop, sediment scoop left, and there's quite a few.
You got Bender? Bender knows a good idea. What? Unless he's watching it. Where's his glasses? I'm gonna show there. What? Come on, glasses. Flying glass. That's They'll get home. Have fun, y'all.
Thank you so much.
Yeah.
They get a lot. We took lots and lots of pictures. Many. Been here for about.
It's old. You want
Sorry.
the torch?
We didn't have like told us not to go, we're just like.
Wow.
You also increase that nice.
There you go.
Stunning.
Looks like a pretty good.
Hello. Afternoon. Oh, there's an egg case. Hi, everybody. Greg Rouse here, joining you from another seep at El Quisco off the coast of Chile. And the team here has been trying to come up with names for these amazing seeps. And yesterday, if you were watching along, we were at a pretty impressive seep site. And the suggestion for that name is going to be Altazor, named after a Chilean poet. And I think the group's all pretty in favor of that. I told you yesterday, if you were listening, there was a clam described from here after Pablo Neruda. We've been seeing that clam shell quite a lot. And the team is trying to think of a name for this particular seep feature we're going to be exploring for the rest of today. And I'm joined by Ana Adamo here, and Fernanda and Erin will be doing the logging. So the four of us will be, with the great help of our ROV team, for the next few hours. So we are transiting to the next waypoint. Would be a good idea. We're at 9. Where are we? Yeah, 9. So we could head on to 10.
Roger.
Thank you. Time here off Chile is midday. People have just broken for lunch. And we're at just over 400 meters, water temperature 7 Celsius, oxygen's not too bad, 100 micromolar, so we've come through the oxygen minimum zone. We didn't get a chance to map this seep, unfortunately, this morning with the high resolution mapping system that's on board. And this is our last day here, so we just went for the dive instead.
Big target on the port side, right?
20 meters up.
Oh yeah?
See the sonar?
You wanna
Yeah,
Take a
let's
look at it.
go have a look. Oh yeah. As with the seeps we were looking at for the last 2 days, no one's ever visited here with an ROV. These seeps were discovered by fishermen picking up on their lines or nets, shells and tubes. And we had no idea how amazing these seeps were going to be at this depth of 400 meters. The tube worm bushes have been especially spectacular. And we might be coming up on another one now.
There it is. Here's a small. This guy is what we saw.
Oh yeah, let's have a little look. Maybe there's some rocks we could pick up for Lisa. Yeah, so here you can see clusters of vestimentiferous tube worms. All the ones we've seen have been what we call lamellobrachia. And in the Eastern Pacific there are 2 different species, and further north. Right now we're not sure what we have until we do some genetics.
Right there. Is that rock on the right?
Yeah.
Does that one look good?
Yeah, let's have a go. We have so far
I'm gonna get you closer to that rock, right?
managed 1 rock,
That's to the right.
and we'd like to get
You put lasers on.
4 or 5 if we can. And Lisa Levin, who you've just been,
That one.
if you've been listening, watching, has been narrating for the last 4 hours. She's now off on break to try and catch up with all the other work that she had from last night and have lunch. And we'll add to her work for tonight if we can get this rock. It's a lovely little mound covered with quite a lot of coral, hydroids.
Tube worms covered in mud. You ever get a poke? See what it is?
You can see the red plumes of the tube worms coming out there. We've seen 2 kind of forms, curly-Q clumps like this and longer forms. We collected both kinds yesterday and we'll be doing genetics on those back in San Diego when I take them back, and we've got some beautiful specimens to put in the museum in Santiago.
Now it's stuck to the ground.
Okay.
So we're gonna look elsewhere.
Yeah. Yeah, let's just keep.
I think I saw some cracked rocks over here.
Okay.
That looked a little too big, but maybe a piece of it.
Flanges are good
See,
if
it's
we
like
see,
cracked.
yeah.
That one, those 3. Oh, it's a bag.
That's it, yeah, let's try. It's
Okay.
cloth. We've seen a bit of cloth.
Alright. Proceeding. What the little one on the red laser there? Is that worth it?
Probably attached, but actually we could try either of these. Maybe they'll pop off.
Okay.
You want to taper forward?
Okay. Alright, let me reposition that a second.
We've had 3 sampling dives. This will be our 3rd sampling dive. We have to leave today, head a little bit south to a canyon. So the next few days will be a little different. exploration. We don't expect, or we don't know. There might be seeps,
There's two rocks on the left.
but otherwise there'll be some pretty interesting canyon features
to the arm out and test those rocks on the left by the
to watch for the next few days.
crab. Right, the one on the left laser.
You don't understand.
Okay, try that.
I'm gonna try and sneak this one more.
Yeah.
Oh, nice. Yeah, that's a good one.
Can you get stable?
Yeah. Hold on. Give me a second.
So while we like to look at all the big animals, there's so much of the biodiversity here are small and we can't really see, so we have to bring them up, look at them under a microscope to really document the site. We've been sampling all of the big creatures, preserving them, photographing them. And we also do that with small things that we can't see but then have to be worked on at night. So this rock will be carefully picked over and the animals identified, used for stable isotope analysis by Lisa. And we get replicates of the rock so she can get an ecological sense of how this community functions.
Photoshop.
That's great.
Okay.
Could be in BB4, eh?
BB4.
Yeah, front.
Roger.
Yeah.
Alright, bring the camera in. Are you ready for the portrait to come out?
Yeah.
Alright, portrait coming out easy. Image bring out a little bit.
Oh.
It's in there.
Yeah, it's right there. I think the bio box caught on the arm shoulder. You see that?
Ooh.
That's what bumped the arm. Was the bio box and bumped that.
Yeah. You want to get another one?
Yes, please.
Just gonna watch that.
That can be a geology sample.
Sorry.
Alright, the camera's back on. I'm gonna go back down. What about this one right here? Left laser there.
Okay. Can you favor to the left?
Roger.
Yeah, this would be a good one.
Yeah.
Okay, let me know when you're stable.
Alright. Try that.
We've got 24, I think 24 scientists on this leg. We started in Valparaiso, coming up on a week ago, and we'll be heading down south to Puerto Montt
November, we'll switch out with another team for the second leg who'll keep going south. And they're going to end up in Punta Arenas in southern Chile, Patagonia. We'll be looking at seeps and canyons in this leg. If things continue to be as spectacular as this, you'll have an interesting time ahead for the next 3 weeks with us. Okay, you could rotate it. Yeah.
I don't want to rotate it.
Drop it. Yeah, got it. Okay, let's take it. Thank you.
Alright, I'm gonna retract sign camera. Okay, yeah, one. Check there.
Okay. Alright.
This leg is being led by Eulogio Soto, professor from the University of Valparaiso. And he has a number of colleagues
Yeah, that's good. That's good.
Okay.
along looking at the animal diversity, also the microbial diversity, physical oceanography, geochemistry.
Correct. Is it 4B? 4A?
4B,
Okay.
yeah, 4A, thanks, Ben.
4.
And he has several students and
Can you zoom in on the SIP?
a postdoc along
That's it?
Yeah.
with us doing microbiology. We have a geology team from Louisiana in the US who've been doing some amazing coring at night. We can't really share the imagery of that, but we're watching this gravity core. Look up gravity core for some images of what these things are and do. They've been trying to punch holes into the sediment with this massive weight at night,
Okay, zoom out.
as well as multi-coring, CTDs, measuring the water parameters.
Yeah.
So it's 24-hour operations here all the time.
Extraction porch.
And joining me today is Anna Adamo, who's based in Norway. And she can tell us a bit about what she's up to. In Spanish, maybe English, maybe Italian. She has a choice.
Okay, get started.
Go back on the line. Let's-
Insieme alla spedizione lungo il margine cileno, è una spedizione dove stiamo cercando di analizzare e trovare delle infiltrazioni di metano e fortunatamente ne abbiamo trovate alcune molto interessanti e nuove, a cui abbiamo dato anche un nuovo nome. Stiamo ancora decidendo il nome, ma sembra che tutto il team abbia deciso di dare il nome al Tesor in nome dei poeti cileni lungo questa costa. E io sono biologa e ecologa marina, lavoro all'università norvegese, alla Nord University in Buda, e l'obiettivo nel mio piccolo studio, come parte di questa spedizione, è lo studio dell'acqua marina utilizzando l'approccio del DNA ambientale per poter effettivamente cercare di caratterizzare gli ambienti gli ecosistemi marini che stiamo esplorando, e cercando anche di paragonare i risultati ottenuti con le immagini che analizzeremo con l'artificial intelligence, quindi con l'intelligenza artificiale, delle immagini della ROV che otterremo alla fine della spedizione. Inoltre, siccome sono siti molto vicini alla costa cilena, dove c'è una grande attività antropica, quindi cerchiamo di vedere anche se c'è della microplastica, e questo sarà fatto analizzando sia la parte dell'acqua, quindi campioneremo anche l'acqua marina e anche grazie al collega della Louisiana Davide Oppo, un altro italiano che però che lavora all'Università della Louisiana Lafayette negli Stati Uniti, lui analizzerà invece la microplastica. Tra tante analisi che farà sarà anche la microplastica nei sedimenti. E inoltre, per ultimo, in collaborazione con un'altra collega negli Stati Uniti, in Texas, stiamo campionando anche quei coralli che riusciamo a incontrare, anche se non tantissimi, ma cerchiamo di campionare i coralli per poi identificarne le specie. Thanks, Greg.
Let's zoom around, just get an overview of it for the record. Thank you very much, Anna. Back with me, Greg Rouse. So I'm also here with my colleague Lisa Levin from Scripps Oceanography in San Diego. And we're both interested in the animal fauna, and we're collaborating with Elohio, our chief scientist, and colleagues ashore on documenting the biodiversity of these previously pretty underexplored systems. So this carbonate mound formed by microbial action with lots of tube worms. The community here is probably mostly surviving based on a methane energy source. The ring of the mound has got a lot of sediment with clam debris. The clams themselves would also be living off hydrogen sulfide, which is ultimately derived from the methane.
Okay. Want to proceed to the next waypoint?
Yeah, let's keep heading to 10. We haven't had that sea star, so could we stop and grab that?
Absolutely.
Thank you. So we're trying to sample as much of the biodiversity here as we can. And that sea star we haven't had at either this side or further south. So we'll take that, get some nice images, and it'll end up in the National Museum in Santiago. Probably, given its size, it's a known one, but you never know.
Sure.
Yeah, there's a cat shark. The lasers, you can see 2 green dots are 10 centimeters. The video can be pretty confusing about getting scale, so these are really handy for us.
Man,
Yeah,
that's
you
cool.
notice how we're not sitting in a cloud of dust?
Cool. Well, we're about to watch.
Pressure's on now.
I want a perfect landing.
I'm gonna try, but
Here we
We're
go.
just going great.
I'm still in the learning process.
Can we go on a quiver?
Quiver?
I think it'll fit.
Yeah. We'll make it fit. You're in such a good mood today.
Wow.
Oh my gosh,
See now when he ramps out, that's when we want to open a quiver.
Yeah, we'll get this shot and then with the quiver operation.
I sit down.
Well, that was a great landing. Beautiful asteroid. I'm not great on sea stars. I've worked on some, but I have a colleague, Chris Ma, who is great on these names, and I'll send this frame grab to him and he'll give us a name later. But feel free to comment on the chat, or someone in our own chat might tell me. We have a WhatsApp group, so we're constantly
14 science channels.
chirping at each other.
Whatever we want.
So sea stars, like nearly all echinoderms, have a water vascular system, and that allows them to separate from the circulatory system, and those generate the hydraulic pressure for the tube feet. You can see those little translucent appendages coming out from underneath the arms, and that's what they use to crawl with.
We're going to start opening the coral cover now.
Yeah, great. That's great images. 1 and 5 are done, so any of the others behind that.
So you can't, yeah, that's come back, yeah.
So Ben will be able to pick this up, the red silicon.
It's pretty small, isn't it?
Yeah, it is. We did get one in the bush of the tube worms yesterday, but I think that's different. You can see a little red eye spot there at the tip of the arm. Each arm has its own. So that little silicon extensions make it much easier and gentler on the animals when we collect them. We'll probably have a look underneath that sea star. There's often symbiotic animals living on them.
It's pretty close. Okay. Nice.
Perfect.
I have 2. There's a little star.
Our current plan is to continue for about another 5 hours. sampling at this site. Julianne, will the BBC dive later be streamed? Or is that just for that? Will stream?
Actually, I'll have to check with them.
Yeah. There might.
We don't stream BBC dives, but I can check with,
Yeah, we'll let you know if there's going to be another dive tonight.
In order for me to retract that brand, the section nose has to be clearer than, like the shoulder, you know what I mean? See how it's coloring?
Otherwise, we've got our transits and mapping. Maybe diving tomorrow. We'll have to let you know about that. We're going to a place called the San Antonio Canyon. Not too far away. Oh, we've got an ID on the Sea Star. Thanks for that. Chris Mars says it's a low faster.
Nice sea star, Ben. Little guy, wasn't he?
All right, thanks for that one. That's a basket star. No, it's a coral with a snake star on it. Okay, we have a request to go and have a look at that coral.
Okay.
Thank you.
All right. Skate.
Got a skate. The large shrimps are called heterocarpus. The small shrimps are different. We've sampled all of those for the museum. And the whelk you can see crawling away from us, the white shell up in the upper left.
Follow the skate. Okay, we'll go to the core.
Let's turn the camera up. There's the camera, straight.
We've sampled it. We've got it. We got it yesterday. Yeah, we'll let that one go.
You want to let the coral go?
No, we'll go for the coral, but we'll let the snail go. It's running away as fast as it can. Looks like this coral's growing on the tube of a dead tube worm. They do need something hard to attach to, so in a pretty much muddy place, this rocky carbonate seep is turning out to be a pretty good habitat for corals if they can tolerate the toxic, relatively toxic environment. Out here, there's probably not a lot of methane flow, so this one's just relying on the plankton drifting by, but those nearer the seep, that's small piece will be sampled for Anna and her collaborators. Do you know the name? Okay, this could be Paragorgia or Leptogorgia.
Is this an octocorp?
Yeah. You can see the tentacles. There are 8 on each polyp. If you can. Beautiful.
I'm gonna sneak your Stobergram out. I think if I put it in the cover, right?
The arm, or one of the arms on the ROV has cutting blades, so we're able to just take a piece, usually, and that'll be fine for the genetics and anatomy.
Okay, Stobergram's out. All right. We'll go for try to snip all of those off. Put in the coil cover.
Yes, please. Yeah, 6. Or anything else?
6.
Yeah, 6 is good.
Oh, those eyeballs of those shrimp
Yeah.
coming at us. I think we've got a bunch of fans in the background.
They really are abundant. It's notable compared to other places I've visited. We haven't got a name on that little shrimp yet, but they had shrimps have a rostrum, a little spine that comes out between their eyes and extends forward. And this one has a remarkable rostrum, quite long. Yeah, go ahead. Thanks, Ben. Maybe then, if it holds well, hold it up for a close-up. Thank you.
Just a little up.
Oh, you can see the big rostrum on the shrimp there on the lower, just beneath the coral.
Is good enough? Great.
Yes, that'd be Anna. Good enough? That's it. Perfect. How do you guys sharpen the blade? Yeah? A honing steel? So it's attached to a vestimentiferan worm. You can see the attachment point down there. So the coral just settled on the random tube. And it's a bit tricky because we don't have the weight of a rock holding it down to help break it off.
It's too difficult, maybe. Because it's too big.
Oh yeah, we won't take it off. Yeah. So these gorgonians have a proteinaceous skeleton. It's not calcareous, so it's flexible inside that pink tissue covering. And as you can see, it presents a problem to cut it off. We can drop it. Should we drop it? Or do you want to use the other arm?
I'm gonna get a better one. If I can follow the camera.
Sorry.
What are you going to do, Ben?
I'm just gonna re-grab it.
Oh, okay.
Maybe a different part of the blade is sharper.
Okay. You sharpen it every night?
Sometimes,
When you remember, yeah.
I think sometimes those materials are just so strong, like glass sponges too. Those are tough.
Yes. Yeah. They have all these glass rods in there.
I mean, yeah. Sometimes even shears don't cut them. They're tough, man.
Yeah, you can't even see where it was trying to cut.
Yeah,
You can see the blade is closed on it there. It's just not able to cut through.
That's surprising.
It's okay. I'll get this.
All right, Ben is determined.
Zoom out.
Pilots love a challenge. Usually when it involves a suction sampler and chasing something mobile, but also this is another good one. Push coring, they're not so fond of. Push coring is a real challenge, as you've seen if you've been watching. Okay.
Okay. The sit camera's gotta give me a good tip. Alright, I could zoom in on that, you know what I mean?
The tube there is of one of the tube worms that we've seen and hopefully we'll see more of in abundance. That tube is made out of chitin that the worm secretes, and then the fuzzy covering all over it are arborescent or branching foraminifera, which are technically single cells but can reach very large size. They're not an animal. Forams often have calcareous tests, but in this case they incorporate sediment to help them give structure. And they're often associated with seeps.
It's sticky too.
great job, Ben. If we zoom in, we might see that skeleton inside. Yep, that's the internal skeleton. And there you can see a couple of polyps contracted going away. Okay, thank you. Can you talk about the goal of these corals or what their future study will be, Anna?
This sample will go to the Dr. Erin Easton in the University of Texas, and she will then analyze taxonomically the species. That's why she will be the expert in this. She couldn't come to this leg, so I'm collecting for her these corals. I'm more expert in Sclerotinia, the hard corals, than the soft corals or octocorals. And the idea is also to understand how many species and the frequencies, the abundance of some of these corals. So maybe in future we can also do some analysis, population genetics analysis. So, but let's see. Doesn't seem that there are so many. So let's see.
Have we seen any scleractinian stony corals this cruise?
I saw one, actually. I heard you say that it was a Desmophyllum, but I couldn't see in the video probably.
Oh, that I saw? Yeah.
But yesterday I saw another one, I think was Caryophylla, because it was different. It was very small. It's another solitary coral. But we don't want to collect unless it's something specific or something very special. But hard coral, we will leave it. Yeah, but I couldn't see so many, actually.
Yeah, I've been surprised. I thought we might see some, maybe in another leg or in the canyons coming up.
Yeah, maybe.
We get the host.
So Anna's well known. You might have heard of Lophelia reefs. And has previously studied Lophelia, which is a colonial deep sea coral. You can look up Lophelia, Lophelia, and then there is Desmophyllum, is a solitary coral. But Anna showed they're all one group, and created a lot of controversy.
Indeed.
Because everything became Desmophyllum, but everyone calls the reefs Lophelia. It's kind of become a common name now, right?
Yeah, they're starting.
Even though the genus is gone.
Yeah.
Yeah.
I mean, it's still used because people is used to call, as you say, Lophelia reef. But yeah, they started to use scientifically the name Desmophyllum pertusum. We also need to say that sometimes even we wish to keep the names, we have to follow the rules, the systematic, the zoological rules. So there is a code that we have to follow. In that case, we couldn't keep the Lophelia name.
Yes, there's a code, and it's annoying a lot of the time, especially for ecologists, or when names are very established and commonly used, like Lophelia. Sometimes we have to go to what's called a senior synonym, an older name. So Desmophyllum took over from Lophelia. And we like to, if you use evolutionary trees as your guide for taxonomy, it turns out in many cases we have groups within groups, and Lophelia is within the Desmophyllum group. So to properly reflect its name and membership, it should be referred to as Desmophyllum.
Updated position, thank you.
And we do this all the time. Sometimes there can be petitions to keep an older name, and there's a commission of taxonomists who rule on this, but normally we just follow the priority, the oldest available name. So we're on the hunt for another mound. That's the radar sonar looking.
Clear. No mounds in this area.
Okay.
Headed for waypoint 10.
Yeah.
Pretty close.
There's a little rat tail. We did get one of those at the southern site that'll be used for taxonomy. We've sampled pretty well, actually. We've got a good, quick overview of the diversity here. Of course, there's always more to discover. At Seeps in Costa Rica, where Lisa and I have worked with other colleagues, we did 5 different expeditions to seeps there and found more than 450 animal species. We haven't done a tally yet on what we've collected here so far, but we'll do that at some point in the cruise.
Some small features.
Where's that? Slightly forward there?
Yeah.
With the shadow?
Oh yeah. Let's have a look. We should use 29. Yeah. Yeah. Okay. We're just talking about logging. So this is now actually dive 729 of Sebastian. We actually went in the water earlier this morning to try and map, but the sub had to be recovered from some electrical issues, so it gets a new number when we go back in. So we're actually on number 729 for Sebastian. Okay, here we are at another feature. Looks, I bet there's some rocks here we can get. Lots of tube worms, some carbonate. You want to try that? Yeah, there might be 2 possibles there. We've had more luck on these kind of lower mounds for picking up rocks. When we get to the big ones, they're just solid. It's still pretty active. There'd be methane seepage here. You can see bacterial mats. The tube worms are happy.
Yeah. A little shark.
Cat shark. Can we see the lasers so you get a sense of your chances? We'd heard there were tube worms here and clams, and I guess in my mind, I thought we might see something like this. A few tube worms here and there, but we've been stunned with the sheer number of tube worms in some places. It's really fantastic, and I hope we'll be finding another one of those big mounds today. That should fit if it comes up.
Gingerly sitting here. Ben, if you want to try the forearm. See it's loose.
This rock's covered with those branching arborescent forams I was talking about. It's got a hydroid colony on top. Hydroids are different to corals. The corals and anemones and octocorals are all in one group. They have 6 or 8 tentacles. But hydroids have a polyp phase, but they also have a medusa phase where they bud off clones of little jellies. Whereas corals and their relatives don't have that medusa phase.
Is that a good rock?
That's great.
Might crumble.
So you want to go straight to the box?
Yeah.
Let's not hold it too long.
Yeah.
It's looking good though.
That'd be smart.
We could go BB1.
Get a good shot. Goodbye.
Okay. That's great. Lots of life on that. You can see the arborescent forams. We've got the little primnoid. Oh no, Swiftia maybe.
Yeah.
And the hydroid. We've got an anemone down underneath. And there'll be lots and lots of creatures in the nooks and crannies of that rock. BB1B on the starboard.
You gotta watch your shoulder there.
Or you could go for 2 if it's easier.
That's good.
We gotta go 2.
4, 2. Okay.
We gotta go wrong way.
That's good. Might be a little too far. Is it gonna clear the door?
Yeah.
Either side, Ben.
Okay. Just watch the shoulder, right?
Oh yeah, I see what you're saying. Yeah.
Watch the shoulder. Okay.
This camera's rotated all the way. You're clear. You're okay.
Camera down the middle. There.
Yeah, you're good. It's really close. You're okay.
Still clear.
Yeah, we're good.
Try to get it down. Greg, I'm gonna drop it here.
You can drop it. Thanks, Ben. I think it'll go in. Yeah, great. So we put each rock for Lisa's study in a separate compartment. So we have 8 compartments.
We'll retract the torch if you're ready.
Roger.
And we'll mostly use them for rocks. So far, that's our 3rd rock today. I think we could get another one.
Oh yeah. Yeah.
There's another one just ahead of us.
Can you use this one, or do you want me to move the vehicle?
Which one's it?
We're ready to put lasers on.
Where are you going? Which one?
Alright, sorry. Here. Here.
Oh, no, no. Oh, no. Which one?
I can hop over to it. Okay.
Now we've had a look for a few days. We've noticed some differences where seeps we're familiar with at deeper. So this is unusual because it's at 400 meters. And we've seen hardly any limpets or small gastropod snails. Normally we see many, many of those associated with the carbonate or the tubes. So that's a striking difference. We haven't seen a lot of live clams. We see a lot of dead shells. The clams must be here, but they're likely potentially buried, and it's just been tricky for us to see. They're siphons. In fact, there's a, well, that's not a vesicomyid open valves you can see there behind the rock. Okay, will this come? No. It's attached.
Yeah, it's attached.
Okay.
Okay, we'll keep looking.
About this one.
Maybe we can try it. I can hop over to this one next.
Yeah, that one looks promising.
Okay. I can get you closer.
This one, yeah.
Hmm. Yeah, this one might be attached as well.
It's crumbly.
Yeah.
Attached. It's not really moving any,
Yeah, okay.
I just blocked his entrance.
Trying to see if up there's a flange we could break, but we need it to be pretty thin.
There you go.
Look here.
Yeah.
You reckon you could break, try and break that?
Yeah.
Or even where the... Is that a flange? No, it's probably just tubes.
Let's look at the big building.
It's all area of interest.
It's pretty thick.
We'll just try to break it apart.
And there's one over there. See that one? Yeah.
Or about the tubies.
Yeah. Maybe that one. Well, not that one. I think we can stick it underneath. That's good. You can try that one. And that was attached to.
Yeah.
We can keep line.
Yeah, let's have a little more of a look. But let's do this again here
Yes,
before we
thanks.
take off, please. So Niskin bottle, we'll take a 5 litre water sample here. We try it at different habitats. We have 6 bottles. So far we've sampled 1.
Alright, so, trying this number 2 here.
Yes, please.
Alright, here we go.
There you go.
Okay. Good?
Did one fire and just not flip? Is that what
Yeah.
happened?
Yeah. See, first one is on the upper right screen.
Yeah, oh, it's
So it's
closed.
definitely closed.
Got it.
But then the flag didn't come on normal, but it
Right.
was too close down the flag.
Got it. Yeah, you can actually see the bottom of Niskin number 1 above the arm there, the grey bottle with its caps closed. It's got a cap at top and bottom.
1 and 2 look good. Yep. Alright.
See that other ribbon? Right? That ribbon far off the first one?
Yeah.
That's two ribbons.
Oh, I see. Got it.
Okay.
It's a very fancy system they've got here. Most ROVs have a trip pull system with cords and leads to a lot of angst, broken cords.
We're gonna fly around, I think.
Yeah,
Just
please.
see what we see.
And we'll probably do a slope here once we get another rock.
Another skate.
Got a skate, keeps cruising around.
Looking for rocks for you.
It's actually a nice feature.
Yeah.
There's a bacterial mat there. Something sitting on top of the mound, maybe just a whole bunch of tubes. Yeah. So there's tall, curly tube worms there, tightly coiled tube worms as well. It's a bacterial mat with lots of tube worms on top of it. That could be a nice image.
Ben wants to poke a rock here.
Okay.
He's talking to me. Drop.
And seeing a rock.
I don't know. You want to get a little closer?
It's a part of a big piece. No, I don't. Try to push.
Alright, see where lasers are right now, Ben?
Oh, yeah.
Right there? What about there?
That's possible.
Maybe give it a poke and we'll see if it's this and then, do some, see.
Okay.
Maybe get closer.
Like this one, right?
Yeah, poke it. Hold on, let me get closer. Alright, bring you down easy. Just give
Yeah,
her
I
the laser.
think we've got a few choices here.
Yeah, just pocket seeps loose. And,
Oh, yeah, that's a good one.
Kinda. My front tubes are resting on slippery rocks. That's what I'm doing. Yeah, you can try. I'm still a little bit slippery. There you go. Looks like you're gonna drop. It's not a huge rock, but.
That's good. Oh, yeah, it's a good rock.
Rogers.
We could go in,
Looks a little precarious in there, if I'm being honest.
could be in BB1 or BB2.
Okay, you ready for me to push it out a little bit? You gotta watch your left shoulder. See that shoulder? It's pretty close to this rock, so it's gonna go to one Bravos. See how close it is?
That is a big rock.
We could do
Should fit. We've got 1B or 2B.
It's 2B. Yeah, you wanna do 2B? Okay. Alright, 2. Right next to the other one. Yeah, I think your shoulder is contacting the rocks there, but I don't know how to fight that. So, see that? I mean, you gotta get over it, so. Throwing outside of it.
There's a challenge to getting this rock in the box, the way the arm is sitting. We've got this basket really loaded with
Yeah, it's okay. You're okay.
boxes, so as the arm is
You're okay.
pivoting, it's hitting one of the boxes. Presenting a challenge for Ben. Yes, please. Beautiful. Okay, that's a great sample. We've got 4 rocks now.
Okay.
Let's,
Pretty unfortunate.
for a little variety, might do a bit of sloping.
Sure.
Thank you.
Take off just so I'm grinding.
Yeah, we could maybe try it. Yeah, let's try it among the tubes over here. the low-lying tube bush. That'd be good. We did a big, long tube bush, and let's see what if anything's different in the smaller ones. So these tubes form lots of niches, crevices, holes where other animals can live. Bacteria grow all over them, and then animals come and graze on the bacteria, and then other animals come to eat them. So, oh, we've got plastic. We'll try and avoid that. So we use these suction samples to get an idea of the small things, small animals that are living here, that we otherwise wouldn't really be able to get.
Slowly coming down. Okay, you want me to grab this or you can land there?
Can we go to chamber number 2, please?
Two's active.
Yeah. Yeah, maybe not watch out for the plastic bin, but
I can move it a little bit away from that.
it's quite a bit of plastic here. So this is a little mound of lamellobrachia tube worms, pretty tightly coiled up together. They're not clonal. They have all settled here individually as little larvae. The females, there's males and female worms, and
Right, Ben. Parked.
the males send out little packages of sperm.
Let me know when you want
Female
something.
picks those up, fertilizes her eggs internally, and then releases little babies, probably thousands, potentially a day. And then they drift around in the plankton, and they have to find a seep to settle on, and then acquire the bacteria that they need to live. So most of them don't make that lottery journey.
What's that purple
This, yeah, that's I don't know, but it's got limpets on it. That's our first limpets we've seen. That's a good target. Behind the beautiful snail.
That's a purple guy.
Yeah, I don't know what it is, but it's got our first little limpet shells.
You want a video first?
We could go tight on those limpets if we could.
I don't have the camera. Go ahead.
Maybe we'll get a better idea of what that thing is. Is it just a random rock? Okay, beautiful picture of the Calliostomous snail there. The beautiful iridescent shell.
Focus on that.
Understood.
We've sampled a few of these for the genetics. Try and get its name. It's probably a known species. We'll find out. And then behind it is
Yeah,
Oh, it's an egg case. Okay. Interesting why the limpets are choosing to be on that and not the tubes. So, thank you. Great view of the crowns there of the lamellobrachia. They're feathery, they're acting as gills, it's actually their heads. And the red is hemoglobin. So they're breathing, they get oxygen through that, and their blood can also bind with hydrogen sulfide, and their bodies are
Lunchtime.
filled with bacteria. Here we've got a spider crab eating. What are we seeing?
I don't know.
Okay, they're sharing it. It's not quite clear what it is. Could be a dead anemone. You agree? Anemone? You think it's an anemone?
Yeah.
It's a clam.
If it's a clam, then we need to find them.
See the crabs coming in?
Yeah.
I can smell it.
Yeah. Okay.
Yeah.
We're going to try and get those limpets that we saw before that are sitting on maybe a cat shark egg case.
Alright, the lipids are on the purple thing over there.
Yes, please.
Yeah.
Yeah.
Okay. Don't turn it on yet. Okay, yeah, let me know when suction. So I want to get behind the shell and get something. Okay. Ready on section. On your mark. Okay. Okay.
Looks like that egg case is hatched out, so we're not risking the little baby shark
Sounds
there.
good.
It's already gone away. There's a scale worm swimming.
Where else do you want to go? Okay.
Oh, you could get that crab, Ben. We haven't had it.
Don't turn it on yet.
New crab.
Alright. Section's off. Okay, turn it on. Section's coming on. Section's on.
Oh, well done. And then just go in amongst those tubes to get the different.
here?
Yeah, that'd be good.
Section's still on.
And maybe more just. Yeah, that's good.
Tube is solid dust at this point.
Yeah.
I can't see what's going on there. There's stuff chunking around in there.
Probably. No, we're good with the snails. Okay, I think we'll call that
Alright, put
enough
clean
for
water.
me to sap.
They'll clean it out.
There's a lot of
There's some stuff sucking
a lot of sorting for me with that.
Put in some clean water for a minute. We'll clean it out. Yeah.
Okay.
Alright. Do you want me to go until the jar gets a little cleaner, or do you want me to leave it?
Well, you could keep running it through.
Okay.
Yeah, we've got a pretty fine mesh on this.
There's another crab.
The same one?
No, yeah, I think so.
Same kind? No, it's okay.
Alright.
One will be good.
I'm gonna kill sections off.
I'm just gonna hold off on this in case everyone wants
Alright, jar 2 has stuff in it. Which jar would you like me to have ready for the next position?
Bubbles coming out of the middle of the two-frame mat.
Oh yeah, let's go to 4. We'll do a coarser
That's
sample
3.
next.
That's 4. Alright, 4 is active. Okay. Sweet. Yeah, there's some bubbles coming out for sure. Basically right where we were rummaging around kind of.
Yeah, so
Yeah.
we've disturbed the flow there, and that's gas coming out.
I'm waiting for the crab to say, my bubbles.
The worms aren't directly using methane, they're using hydrogen sulfide as their food source, which is a breakdown product of methane.
Okay.
Okay, I
Would
think
you
we
like us
can
to move,
head on.
Do you want us to keep the section sampler active or stow?
You could stow it.
We'll
Yeah,
stow
I'm
it.
not sure what's next.
Alright, let it take off. Let me stow it in here. Alright. Yeah. Alright, we're gonna blow it out.
So we're just offshore from El Quisco, a town near Valparaiso in Chile. We can actually see the shore, so we're not far away from the coast, and we're in 400 meters of water. So this is a pretty accessible seep for people in the future if they want to keep studying it.
I took it from you. It's alright. That's the Google have control.
Earlier today we saw a wonderful
Yeah.
tube worm aggregation with a
Alright, starboard rim track.
lot of fish that are commercially caught. This place is not a marine protected area,
Alright.
and I'm sure a lot of this imagery and sampling that we've been doing here may lead to a reassessment of this site.
Yeah, that's alright. There's another mound I can hop to. You're ready.
Yeah. Oh yeah, go for it.
10 meters on.
Let's see.
Okay, proceed to the next. So we're target here. And we'll go that way.
We've got a suggestion in the chat that the little boxy crab that we just sampled, first time we've seen that. It's called Platymirror, so we'll note that and when we photograph it tonight. So thanks for that.
It is.
So we've just been mound hopping this whole dive from mound to mound.
See the next mound too.
Yeah.
What do you see?
Yeah, the long tube worms, coily tube worms. We sampled enough of those to figure out what's going on. Up on top, we've got a beautiful basket star. We've had that fish. Yeah. Fish. Yeah, we don't want to go near that. We learned our lesson. Yeah, it's a curled up hagfish underneath there. That squat lobster has a very long claw. I don't think we've had that. It's only got one. Yeah, that's a beautiful basket star. Yeah. You can have a look. So coming into the frame is a beautiful basket star with its, we often see them perched up high, their arms unfurled, catching plankton. So it's a kind of brittle star where the arms branch. They have lots of hooks on those arms to catch little things like copepods and small shrimp, mysids. Yeah, there's a great view of those arms. So they have 5 of those, and it's holding on with some of them and just stretching up some of the others to try and catch food. We caught a beautiful specimen of this yesterday, so we'll leave this one alone. Yeah, we can keep going. We've only got our one dive here today before we leave, so we're trying to get as much of the site looked over as well.
To hop to the next mound?
Yeah, let's go to the next mound.
It's kind of dead down. So that was just waypoint 10 from C to 11. Yeah, dead ahead.
Anything interesting with the methane, Scott?
No, it's elevated a bit, but nothing. Let's put it this way, Greg, it's a nice high baseline.
So we have an instrument on board that's measuring the methane in the water. And Scott, who's along from Woods Hole Oceanographic Institute, is monitoring the methane levels. It's fascinating to see the methane.
Do you want to sample this rock?
Huh. Is it a big tube bush? Or just an enormous rock? It's got a coral on it. It is just a rock. Yeah, we should have a look at it. So I wonder how that was formed.
It's a nice boulder.
Yeah, let's
Can you put that at the front? Maybe.
Hey Dad, this could be a good photogrammetry target. Nice discrete structure.
Yeah.
It's quite beautiful with the
It is great.
corals, anemones.
Dan, do you want me to put the photogrammetry up on the screen or just?
Can we zoom in on this? See what those clear things are?
It's a nice coral. Do you have a bit of sampling to do?
We could sample after.
Yeah, okay.
We'll first decide if we'll sample, but
You can sample the other one.
or it takes a while to get ready.
It'll probably take 15 minutes just to get everything up and running. So do you want to have a look around and see if there's stuff to sample?
We can come back to here. So yeah,
okay.
let's do that. But we'll have a look around. So we've got a camera rig on board that will allow to make 3D models. So this is a pretty good sort of structure to do that on. And we'll leave this intact and come back to it. But we'll have a look around. See if there's anything in particular we might want to take. These clear structures, what are they?
Pretty small.
I think that's tunicates. So this is a colonial sea squirt.
Oh, like this is round.
There's a beautiful polychaete poking out, a unicid. So tunicates can be solitary, or in this case colonial, but still it's a clonal group. Every individual has 2 openings. You can kind of see them opening and closing as you look at the different ones, and they take in water through one hole, filter it, and pass out the clean water through the other hole. It's the first one of these we've seen. Okay, let's have a look around, see what else we got.
This is one.
It's a nice sponge there, the yellow, orange. Also, don't think we've seen that.
And then all sorts of stuff.
Quite a lot of amazing things on this rock.
His arms are like
Lots of different octocorals.
dirty.
Yeah, that,
They're dusty. These guys have oversized arms.
yeah, we picked up a smaller one with the hairy arms like that the other day. Now I realise, but we haven't had an adult like that. It's impressive.
I know Chile squat lobsters mentions. Might put minis just about on those little rock. This is the same too, I think.
Yeah, that's another one from the side. Sea squirt, tunicate. It's a stunning sea anemone there. We've picked up a few of those living in the tubeworm bushes. We've had this coral today, and we've had the white one lower down.
Like giant fish.
Yeah.
Good.
Can we look at the coral to the lower right?
Which one? Right here?
Just that colony there.
Oh, the big white one?
Yeah. That one?
Yeah, that one, yes. But I think that there is another one that is behind that maybe.
Can you come and point it out? So Anna's just going to point out a potential coral we haven't sampled. Oh, over there.
If this is for the photogrammetry, maybe better than
Okay. Yeah, let's leave this rock for the moment and we'll go and have a look at the coral.
Understood.
Thank you.
Hop over to the ground plane.
Straighten your camera.
So this rock's about 3 meters across, maybe 4, 2 meters high. So it's a pretty ideal structure for doing photogrammetry on it. There's no obvious seep animals on it, so maybe this was formed and somehow no longer in any flow of methane, and now it's just serving as a reef.
Can we zoom in on the camera?
What do you think?
this one.
It's different?
From
Or we-
the one that we collect, yes.
Yeah, okay. So we'll take a piece of this one.
Cool. We'll get you a little closer. Yeah.
Is Photogrammetry can powered on?
Give me a second.
So the last 2 days we're about 1 kilometre south of here, exploring seeps down there that were very impressive.
Is it starboard now? Push down a little harder. Stationary.
We'll probably give those the name Altazor.
Did you get a good enough close look, or do you want one more close look before we zoom?
Anna, how's
Yes,
that?
close. Yeah, thank you.
Do you know the name of this one, Anna? No.
No, I'm chatting with the expert.
Okay, Anna's trying to get a name on it for us. Okay, let's snip a piece, please, Ben.
With the back four open, or the lids is already on three. So yeah. Back to it. Yeah. Oh, sorry.
See that, Rick? See it?
This thing? Yeah, I don't know what that is. Egg mass? Maybe eggs. Like a snail egg case. But if it's something you can grab, that'd be useful.
Right. Are you guys happy with the
But
video?
but if you want to just go up towards the tip of branch, that'd be fine. Yeah, we're happy with the video. How's the methane value here, Scott?
Same as it has been. It's just an elevated background.
Elevated background.
No space, yeah.
Okay, the coverage of
Yeah, it's right there, cover's right there.
You want that piece, Greg?
With the eggs on it?
Yeah.
If you can get to that, fine. That would be great. So Ben noticed something stuck on the coral, which a snail might have laid. We'll see if we can get a bonus sample as well as the coral. Yeah, I think that's a whelk egg mass. We've seen the white whelks roaming over the sediment. Do you think you can get it?
Oh,
Oh, yeah, I can definitely get it. I'm just wondering what the best cut
but
is.
it's not egg. Yeah, it's eggs. A lot though.
Because I could take this whole, let me get this off the brush. You got your sticker, your pointer?
Yeah. Oh, that would be
Yeah.
this whole branch?
Yes.
Oh,
No, no, don't worry about that. You could take a smaller piece. Thanks.
tagging. Greg, we are taking the eggs too. Great.
Keep the egg in the,
And-
keep that wings.
Oi.
Oh,
Oi,
look at that.
great.
Wow.
Thanks.
Fantastic, Ben. Thank you.
So they suggest that this coral is paramucide.
Do you want a close-up of
Oh,
the way it is? Oh yeah, this is nice. Paramucide?
they suggest this. Okay.
And we're going into number 3.
Number 3.
It's intact.
Oh, great.
That's all they.
Good. Great, thank you, Ben. All right, so I think we'll spend a little time and make a photogrammetry flyover of the nearby boulder.
Sure. Oh.
Does our audience see the photogrammetry camera, Sophia?
Almost.
Almost? They're going to? Okay, great. So, stand by people. You're going to see something a little unusual. We've got Dan and Pedro along with us from the BBC, and they're trying to get footage for an upcoming series, Blue Planet 3. And if you've been, it is pretty spectacular here, so hopefully they'll get some great images. But another flyover with this special camera system that'll then allow the stitching together of the images to make a 3D model. So when we see a structure that looks appropriate for that kind of model, we'll try it out. And this rock covered with those beautiful corals is an ideal kind of feature to do this. We tried it yesterday over at Tube-worm Bush, but we had so many crustaceans in the water that maybe the model, haven't seen the model yet. Those likely disturb the model. Here we don't have that problem today. Just trying to set the lighting, and then the idea will be fly around it at a relatively constant height above it, backwards and forwards and over and around. And separate frames will be taken, and with reference, hopefully as they overlap, they can all be stitched together. Lasers off.
Don't touch the rotator. Closer.
Okay, here we go. We're starting our approach. It's a bit of flocculent. Actually, they're all little mysids again in the water, but it's not as bad as yesterday.
It's hard to paint up now because the camera's looking. Does that make sense? Yeah. Like this here is not changing. I'm going up and down. Yeah. also like different.
You can see behind this rock there's obvious active seepage with tube worms, clamshells, and Scott tells us there is a normal level of methane that we've been seeing pretty much the whole dive, so it's just amazing there's this rock sitting here. Looks like it is carbonate, which would have been generated from methane seepage. Scotty trying to get in on the mapping action, could end up in the model.
How many times can you put it together? You just never go back on track. I'm probably on the ground. So I'm on the ground.
How many turns?
Yeah. So like, you guys never know. Alright, so I'm on the ground. Yeah. Like, I should feel it come up, come back. It's up to date. A bit more coming down.
That's what I've written out, but I come over that.
Got a watch change happening now. The pilots are just changing out while the photogrammetry continues. Our friend the skate is back.
There is a tab that I kind of which is nice. It's a close the windows.
So in about an hour, I'll switch out and Oilohio Soto, our chief scientist, will come in and, being bilingual, will narrate in Spanish and English. And he's been thinking about a name for this seep site. He might honour one of our Chilean scientific colleagues naming this area, but I'll leave him to tell you that. And to finish up the sampling for this dive, tonight, hopefully, the BBC will get a chance to do some filming. And then we're going to transition to canyon survey. Right now we're about 15 kilometers offshore from El Quisco, and then we will transit tonight. There's a long canyon, San Antonio, a little south of us, that heads out to sea. And right now we haven't quite figured out where we'll go in the canyon. We've got a couple of dives planned, so we'll have a meeting later today to figure that out. But hopefully we'll be diving again tomorrow morning, Chile time.
Just with the ground. Your way. So you said this face one better? Yeah. Okay, over this.
So we've been round the feature now. Interesting how the carbonate is fractured. There's some seep animals along the margin underneath, but there's nothing above.
Yeah. All right. Have you done the photogrammetry?
All right. We look forward to see the results. Hopefully they'll be shared. Schmidt Ocean Institute's very good about open access and data sharing.
Lowers off. You can rotate, sit back down. Rotate up, rotate. Sorry.
Afternoon, Doug. Good, thank you. I'm joined now by ROV Supervisor Doug. And there's a desire for a couple of the corals on this mound.
On this mound?
Yes, please.
Okay.
And then we'll head away. But Anna will tell us which ones you got. Do you want to point out what you like? Here's this. Oh, where the basket star is? We have that.
Okay, so it was B-type. Now we have to turn like this.
Yeah, just give it Doug an idea and he can go closer.
I mean, we can try to collect
That
this
one?
Yeah.
Okay.
Otherwise, if in our way we saw another one, okay, but I remember I saw one here, but we are very close.
Okay, so there's only one kind extra to get here?
No, that one, I want to get one.
Just that one?
Yeah.
Okay.
And the other not similar, so
Okay.
we need to use one that's easy to
Okay, Doug, we'll have a look at this. That's the one, seems to be the, that one.
Yeah.
So we've been sampling as many of the different kinds of habitats here to try and characterize this region, and we'll be doing that further south as we go. And so we have a lot of different ways of sampling. Trying to get things from microbes in cores, microbes in rocks, the rocks themselves, for geochemical studies, and the animals. And we're after the little animals and the big animals. And so we can suction them up, we can take clips and put them in a special tube, put them in a box. Seven. So we'll be taking a little clip of this coral and putting it in a little tube. Happy? Yeah, let's go ahead. We don't need the basket star, so maybe the lower, that little part would be good. Thanks.
Pretty much from the same template. So yeah, absolutely. You have a copy, right? Over here with basket star, but not the basket star. So you just to the right of it there, lower right, just a little bit. Right there.
This one down here is different than the one on
Oh, it's two,
Yeah, it's two different ones. So
but they're the same.
the same.
Same kind.
Kind of.
So, yeah.
Yeah, that one you can see it's right there. Still happy with that?
Yep, that'd be fine.
sorry, I missed.
These corals, as with all the other animals we're collecting,
Yeah.
well, I hope, mostly, certainly anything I'm associated with will be sequenced for DNA to try and understand if it's new or an already known species. Even if it's already known, it's often useful to have DNA data because it helps us understand species ranges. And if we sample enough, we can often do connectivity studies in terms of population genetics.
hold up. Are you happy with the initial picture, or do you want another?
We could get a close-up, even though it's closed up. There's some details on the polyps that can be visible.
Light, black background.
Great. There's lots of little amphipod crustaceans roaming over it. Yes, thank you. That's great. Okay, good to go. Thank you.
Thank you. Buen caprari.
It's gonna fall right in.
Great. We're doing pretty well on the corals today. That's our 5th sample. So Anna will be busy tonight. If you were watching yesterday, we did have a bit of an incident with a hagfish, which grossed us out. On recovery, slime in quite a few places.
Still everywhere.
Did you guys have a lot of cleaning up to do?
They were doing that when I left, Jim,
Oh,
but I'm
okay.
sure they did. There's still some on there.
Yeah. It was in some of the samples as well.
I'm sure it was. It was everywhere.
You can see some interesting videos online about what hagfish can do with their slime. It's a great defense mechanism. All right. Thanks so much.
All right, what's next?
Let's keep heading to number 11.
Okay, we're finished
Thank you.
here.
Yep.
All right.
You happy here, Anna?
Yes, thanks.
Okay. We made a series of waypoints, and we'd like to try and visit them all today. This will be our last visit to this region.
Yes.
Although, sorry, Doug, while I look here, I'm just wondering if we might be able to pick up if these rocks are broken up enough to pick up. Could give Lisa a nice background rock. Looks pretty fractured.
Yeah, one of the ones on top, maybe, mostly.
I think they're loose.
A couple of these look like they might be loose.
Yeah.
Up around That's probably a rock box, I would say, is that
Yeah.
correct? Yeah. Put that in BB1B. Biobox. Biobox, sorry, yeah, biobox, not rock box.
These out here.
Yeah, just to your left. Yeah. Right, where, well, I guess any of them.
Anyone?
Those two were that squat lobsters in between the two, I thought those were pretty loose, but whichever one you begin with. Yeah.
Oh,
any
yeah.
of those are-
That's-
they're all
You want that one or
pretty
just
loose.
touch?
That one you touched is good.
Alright.
Yeah. It's a decent size. Bigger would be better. Oh yeah, the one you touched is good.
Okay.
Yep. Great.
And beauty shop?
Yes, please. Got a couple of different octocorals on there. Some of the branching forearms too, so there may still be some active seepage here.
And the other side or just this side?
Oh no, that's fine. I don't want to risk you dropping it, so that's
Yeah,
that's
yeah.
good. Yes, thank you.
Okay.
And, of course, hang on. Definitely hitting something. Yeah, but, yeah, we're hitting that rock box, you see how you got it cocked open there? Gotta move back towards the left, there you go. See how nice and moving now.
About all in all, just a little bit.
Okay. Camera's back. Alright.
Great, thank you. BB1B,
One bit.
rock number 5.
Is over down the way.
Okay, thanks, Doug. Now we could head off. Let me know when you want another nisket. Let me know when you want another nisket.
Ah,
They usually fall down.
I didn't-
Fine.
I should have done one now.
mm.
Could do one now.
Yeah, close now.
We don't have long.
Yeah, close by.
Can we trigger a nisket, Doug, as we go? Please.
That's three.
Okay.
I know the flag got popped out, but the other one did, so.
Yep. Okay. Thank you.
Yeah, three gets blocked by the cars. So
Oh,
it's like
okay.
4 or 5.
So we just took another 5 litre water sample there. Could be used for eDNA, might be used for plastics, multiple possible uses. Now we're heading along to another waypoint. We're keeping an eye out for mounds with the sonar.
Hey, on the bridge, updated waypoint.
Update, thank you.
We're about a kilometre north of where we were yesterday. Which, if you watched, was a pretty spectacular place, but we've already seen a visually similar mound, a couple of metres high and across, and we're hoping to find some more before the end of today. Coming up on another mound now. Lots of lamellobrachia tubes, vesicomyid clam shells. Just have a look over this mound, see if there's anything worth stopping for. So it's definitely active. There's lots of live tube worms down there, lots of shell debris. Can we try and get this crab, squat lobster rather? It's quite long arms, and we haven't gotten that before. Do you think it's a slopable one?
We're gonna give it our best shot.
Okay.
They're slippery little suckers.
They like a challenge. 10. It's pretty big. Okay. Let's we'll keep an eye out for a smaller one. They're not really possible to grab, so our best weapon or tool is a slope sampler, but that would be too big for our hose. Wow, quite an impressive feature coming up. It's a lot of tube worms. Discarded can.
Can soup.
So Scott's going to let us know how the methane value goes once he gets a reading. So we've seen a series of these very impressive mounds, carbonate. Yeah, let's just have a flyover. So those tube worms are easily 1 metre long, and their tubes probably go down into the carbonate, so they could be getting much longer and thinner even inside the rock. And we've seen quite a lot of these long forms, over 1 metre long, and then lots of others forming these tightly curled clumps.
See a lot of like bluish gray back there.
I wonder if that's a big cloth again. We've seen cloth today.
Okay.
Yeah, I think it's human, anthropogenic. How's the methane, Scott?
Starting this way.
Starting to go up? Yeah. Not surprising. I mean, having so much of these animals here is an indication of methane seepage. Can you have a look at this? Could be a sea urchin. I think that is a sea urchin. That's our first sea urchin here. Okay, I think we'd like to get that. Please. Yeah, quiver? What do you think? This is the famous quiver set that fell off a couple of days ago. And fell off again. And then finally we got it. So it's good to see it back and being used today. It's likely we'll fill it up. This is our 7th quiver sample. Very well done.
Picture?
No, it's okay. You can put it straight in. We'll get a nice photo on deck. Got a suggestion that this is Arbesia spatuligera. Thanks for that. Been sending the sea urchin pictures to my postdoc Nico, former postdoc, now working in Argentina, and he's been helping us out with the IDs. And we'll be sequencing the DNA of this one.
Yeah, you're definitely over it. It's hitting the other edge now.
And it and all the other specimens we've been collecting on this cruise will be used for genetics or other studies, but the specimens themselves will end up at the National Museum in Santiago.
Oh, no. It's right on the edge there.
It's trying to get away.
I don't know if you're gonna get it or if you need to move the hose. You got the reach.
Scott's trying to get down there and get that.
Got the T4 cameras all you got, buddy. Yeah. You can see where you're at.
It's perched on the... You can't see what we can see. There's a gap there.
There it goes.
It's trying...
It's just off to the side.
It's on a metal bar with space next to it, so it's possible it'll fall down.
Nope. Boy, he's right in there.
Is there?
Oh, he's pushing through. See it? There it goes.
It's fallen down.
Okay.
Now it's on the seafloor. We might be able to get it.
I think he went all the way through, so let me
Yeah.
just back up and we'll just get another grab out of
Yeah,
there.
I think it fell on the ground.
I think you can see on the end, it's right under there, so, alright.
Awesome.
Just keep an eye on it, because I'm trying to just handle that down. Trying to keep some. Just out of
There's just some bubbles on the sick cam.
He's up here. Okay. I'm trying to give you the most, I see it now. Is that a good spot for you?
You know what I'm gonna do? Take this hose out of the way.
That's our suction sampling hose there that was blocking Scott from putting that urchin in the tube. We've really loaded this ROV down with sampling equipment, and that presents challenges for the pilots when they're trying to put samples in different places.
Well, at least we can see.
Meanwhile, the crabs decided that maybe the urchin is a meal.
Now it's caught on the top of the arm now. Sorry, I'm gonna get you over there.
On our first dive a few days ago, now about a week ago in Lamari, a bit north of here, there was no seeps, but we did find several different sea urchin species at about 1,600 meters. But this is the first one we've seen here at 400 meters.
Got it, not time.
Great.
Try and brighten that up a little bit and see if you can see that handle. How's that?
Great.
Can we date like that? Yeah, it's fine, because we'll be taking it off if we need it anyway. Yeah. I'm gonna try and sort that suction sampler, and go to fly up and let it drop down, or go around on the correct side, but I think I got it. Okay. Yeah, just hold on to it for a minute. Once it clears, you can stow it, or that skates right up and down. Wow.
Hmm.
Oh, don't go back there. No.
Dangerous back there.
Okay.
It's safe. It's a skate. It got away. Propulsion's back there, and even though it's guarded,
All right, Scott. If you
some
want to
fish have
stow
met
that,
unfortunate ends.
Just kind of floating about. I didn't want him to get go the way of Squidward this morning.
Oh, you hit a squid this morning?
Well, I don't know. I didn't necessarily hit a squid. We were stood still, so
Yeah.
that can be argued if we hit it right at
Yeah,
us.
or it hit you.
Either way,
Yeah.
they fed the rest of the animals. That's as far out as I can go, Scott.
We could, since you've got it out, Scott, we could do a bit of a slurp
All right, coming in with that starboard ram.
just amongst some of the tubes.
All right, anywhere in particular?
One of the tube worm masses.
All right.
Yeah.
We have lots of options on such a jar, looks like.
Okay.
Yeah. Oh, yeah. I think that region there would be
Okay.
good. Yeah.
All right.
And the idea would just be get in amongst the tubes,
Yeah.
not down into the sediment, and we'll just be trying to get
Okay.
what's
What
living
jar
on the
would you like?
tubes. We're on 4, I think.
We are on 4. Yes.
4 is good.
Okay. All right, Scott, just this cluster here, and we get lined up with that.
It does seem to be some shimmering water around here. Or was that just our camera?
We saw bubbles. Yeah, I mean, this sort of shimmer there.
Yeah.
Yeah.
And up underneath where we're about to do
Okay.
the suction.
Yeah, something's going on.
Good spot for you, if you want to move over, let me move over. I'm gonna square it up for you, sorry. Not a good.
Is it shimmer?
I don't know. Do you want to sync the temp probe in?
We might. Yeah.
Yeah. And Greg, methane is rising, but it's not shooting up like at some of the other mounds.
Okay. We just thought we could see some of the water shimmering a bit, which would be an interesting indication of flow. So we might do a temperature measurement here in the sediment after we've done some suction sampling. The temperature here is 7.7 Celsius. So we'll be interested if it may be elevated.
Going in.
Yeah, that's a great area to go in and slope. Thank you.
Ready?
Ready?
Suction's on.
That filled up pretty quick. There was some stuff in the hose.
Could have been a little bit.
Yeah. Yeah, that's great. So there's likely to be lots of little animals in there. We were expecting a lot of snails. Oh, he just got us a squat lobster. But we've seen very few mollusks on this site. The snail? No, we've done the snail. We've got plenty of that. Thank you. I think we're probably good. Oh, the snail's gone in. The squat lobster's gone in.
All right, Scott.
Yeah, we could just let that run through and index. So that was suction sample 4.
Oh, you get it.
All right. Thank you, guys.
Yep, I'm standing by, sorry. That's Scott. Get it out of your way. There we go. All right, great.
Yeah, let's keep flying over it and head to 12. Looks like there's a lot here. So those long tubes you can see, those are the vestimentor from tube worms, but more isolated. The white are clams, shells, vesicomyid clams.
Hey, on the bridge, updated waypoint.
Most likely called Archovesica nerudae. Neruda-I, excuse me. Named after Pablo Neruda. So both the tube worms and the clams need symbiotic bacteria to live, and they're using hydrogen sulfide as their food source. So this is a pretty extensive mound.
Yes.
The white mass of the other lamellobrachia. So it's all lamellobrachia, but they just seem to be in different morphs. Long, straight tubes or tightly curled clumps.
Did you see something interesting or just that bag?
Yeah, do you want to look at that? If we can. Couldn't quite tell if it's animal or plastic. this thing? I think it's an animal. Sea squirt. So we saw one on the rock earlier.
What is it?
It's a tunicate filter feeder. That's a colonial one. I think we'll leave it.
All right.
Thanks, Tim.
Okay.
So it looks like we're passed over that mound. We're on our way to the next one. I think that's one of the tube worms just
Is it?
poking up out of the sediment.
Huh.
Yeah.
Interesting.
Yeah. Probably not alive anymore. We've still got 2 more rock slots if we see a rock that we can pick up. There's a hagfish, dreaded hagfish under that rock there.
That one today. Some garbage in
Yeah, that's a bottle,
there. Yeah. All right.
plastic bottle. What to do, heat pro?
Oh, yeah.
It's like there might be some
Maybe
rocks.
go back to her.
Yeah,
Stop.
we could.
Just keep going towards 12.
Oh, we haven't gotten this thing. So that's a hydroid. And that we might be able to push call.
Is that right?
It's very delicate. I wouldn't want to try and pick it up.
We'll have to back up, sorry.
So we've talked a lot about hydroids and their branching colonies of polyps, but this is a special hydroid that is solitary. It should be pretty spectacular once we get a close-up. So it's a single polyp, and it would bud off little medusae that would then float around as little jellies. Branchiceranthus, I think, is the group. It'd be nice if it faced towards us so we could see inside the polyp. These can get up to 1 meter tall.
Really?
Yeah, amazing. Falcor saw one back in off Australia. It was huge. It came up. It was this long.
Oh.
They were able to grab it with the manipulator. This one's being a bit inside. Oh, this. Yeah, I don't know. Maybe it's coming in from the inside, poking out through the tentacles. So on the inside of that ring of tentacles will be the mouth and potentially the reproductive organs where it's budding off the little medusae. Just wanted to swing around a little more. There we go. So you could tag this solitary hydroid. Okay. I think. Can we see the lasers? And we'll just get a scale and then decide if we could push core it. I think it should go into push core.
So, Scott, your choice. Just start on these two. I'll leave it, if we want to give any of these, if we want to get rid of these, however you want to do it. Okay. Porch out a little bit. Actually, let's go with one of the front, so we don't have to porch out so much. All right, okay. Now, porch. Nice and easy. Anything clear? Everyone's over? No? Yeah. Okay. That's pretty good right there. Yeah, we'll go for 4. Okay, going for 4. Porch back in.
So this is a cnidarian. It's not an anemone. It's a hydroid. It's a solitary form instead of a colony. And looks like it's going to be a pretty good fit for Scott inside the push core. And wow, look at that. So it probably doesn't have a long root down very deep.
Okay. But it goes straight down,
Yeah, you can just push straight. Shouldn't disturb it too much.
Out there.
Nice. Let's hope it all comes up. Excellent.
I'll see porch, Scott.
Okay. slipping a little.
I was putting the boards right under it, buddy.
Yeah, you're good.
Fantastic.
Yeah, you got it.
Okay, wonderful. Well collected. And that's a good sign for me to switch out, and I'm going to hand over to our chief scientist, Elohio Soto. Hopefully talk to you tomorrow.
Great, Dr. Tyler, stop, please.
Stopping here.
He wants to.
He said no.
Okay,
I'll just leave
sorry.
it. I'll catch up.
It's got the small steroids.
Okay. Where are you?
Okay.
Good. Yeah, good, thank you.
Okay.
Heading towards waypoint 12. Hey, John.
Can I say hello?
Buenas tardes, les habla Eulogio Soto, jefe científico de esta expedición, biólogo marino de la Universidad de Valparaíso. Nos encontramos en el sector de El Quisco, a 400 metros de profundidad, estudiando ecosistemas quimiosintéticos, en particular estas filtraciones de metano que se encuentran caracterizadas por la presencia de distintos organismos marinos, como por ejemplo mantos microbianos, fondos sedimentarios anóxicos y también reducidos, pero hemos podido ver también una importante biodiversidad, la cual está asociada a rocas carbonáticas, también a aglomeraciones o agregaciones de gusanos tubícolas que permiten y que dan la posibilidad de hábitat, alimento y otras funciones para una importante cantidad de invertebrados como corales, como crustáceos. Hemos visto una diversidad importante de cangrejos, de camarones. Durante la mañana también fue muy interesante poder ver en torno a estos tubos de gusanos cómo nuestro apetecido congrio colorado y congrio negro buscan ese hábitat para posiblemente refugiarse, descansar o alimentarse. Estos fondos, principalmente formados por sedimentos, rocas, en diferente grado de evolución, También presentan, por ejemplo, la presencia de rayas, de tiburones y también de diferentes tipos de peces. Nos encontramos en este minuto visitando diferentes puntos de interés dentro de este seep o filtración que está en el Quisco, y hemos estado durante el día de hoy recorriendo, descubriendo y observando las diferentes características del fondo marino, lo cual nos ha llevado muy buenas sorpresas de poder observar de forma directa y sin el menor impacto de perturbación, gracias a la tecnología del robot Sebastian que tenemos acá en el Falcor, el barco del Schmidt Ocean Institute, poder ver en detalle los diferentes organismos que caracterizan este tipo de ecosistemas. Se han realizado recolecciones de organismos, se han recolectado también diferentes tipos de especies de interés de acuerdo a los objetivos de cada grupo de investigación, pero principalmente también se le da mucha importancia a la observación de aquellas comunidades que habitan en torno a lo que son los mantos microbianos, estos tapices de bacterias filamentosas en algunos casos que se observan de color blanco, en algunos casos naranjo, y que nos permiten apreciar un tipo de vida totalmente diferente. Ahora vamos a hacer un close-up de uno de los organismos que vemos ahora, que es un coral. Que saludamos a quienes nos están siguiendo, tanto en el chat como en la transmisión, y nos escuchan de diferentes partes de Chile y el mundo. Saludamos al Centro Costar, al Centro de Observación y Estudio del Ambiente Marino, Centro de Investigación de Excelencia de la Universidad de Valparaíso, al que pertenecen algunos de los investigadores que se encuentran en este minuto participando de la expedición. Así que saludamos a los investigadores y al Centro Costar de la Universidad de Valparaíso. Se encuentra junto con nosotros ahora en el centro de mando del robot la bióloga marina Fernanda Orrego, quien ella nos apoya con la anotación y el registro de todos los organismos que se observan durante la transmisión. Es un trabajo muy importante que permite documentar y registrar los diferentes organismos, ya sea bentónicos como pelágicos, las condiciones y también las muestras que estamos obteniendo. Mencionar también que Fernanda está cumpliendo un papel muy importante en este crucero, ya que ella está a cargo de los estudios de las comunidades zooplanctónicas a través del estudio durante lances de noche, para poder describir y conocer qué tipo de comunidades, en este caso del zooplancton, habitan el fondo y la columna de agua en torno a estos ecosistemas de filtraciones de metano y ambientes reducidos.
Un trocito.
Piso.
I think it's maybe could be hard. Let's see. Any part, yeah, I think so.
That's a piso. Piso flat.
¿Qué?
Seremos una botella también. Tenemos que
Después de que saquemos la muestra, una botella.
o si no, esperamos en otro sitio.
Oye, ¿querés que saludo a Jeffrey? Por la bendición. ¿Querés que saludo a Jeffrey?
Ah, no, no tengo ido. Pues, decídenselo.
We can put him in.
Yeah, might as well.
Really squatty in there.
As soon as you break that section. Stuff done. There it is. Okay.
There's grass and everything else.
Just because it can.
También les queremos contar que, bueno, terminando las inmersiones en el Quisco, nos dirigiremos muy cerca, un poco más al norte, para explorar e investigar el cañón submarino de San Antonio, o también el cañón del río Maipo. Existen muy pocos antecedentes, tanto oceanográficos como biológicos, de los cañones submarinos en Chile. Por lo tanto, es una gran oportunidad poder estar en este crucero, lo cual nos permitirá filmar, conocer y muestrear este cañón submarino que es totalmente desconocido. Uno de los objetivos principales de este crucero justamente es poder caracterizar y describir la biología bentónica asociada a cañones submarinos y poder compararlos con otros cañones submarinos totalmente conocidos que tenemos en el sur. Por lo tanto, resulta de vital importancia poder realizar levantamientos de datos y obtener muestras biológicas, así como mapas detallados del fondo marino, sobre todo de los cañones que tenemos frente a Chile. Bueno, hace unos instantes, quienes están viendo la transmisión pudieron ver la recolección de un coral a través del brazo mecánico del robot Sebastian, del ROV. Estos corales, bueno, serán investigados a través de la colaboración con la científico italiana que trabaja en la Universidad de North, la doctora Anna Adamo, que se encuentra también en la sala de transmisión y que nos va a dar un poco más de detalle respecto de eso, lo que ella está haciendo.
Hola a todos. Sí, estos corales vamos a hacerlo en colaboración con Erin Easton, de la doctora de la Universidad de Texas. Vamos a recolectar esto. Ella irá bien la parte sistemática y taxonómica de los corales. Está muy interesada en octocorales y también en los corales negros, como lo que acabamos de recolectar. Y yo soy interesada más también a ver la parte de la caracterización de los hábitats y ver si efectivamente detectamos también la señal de estos corales en el agua. Por eso que hacemos una colaboración también con esto y vamos viendo también si en el futuro podemos la conectividad, la dispersión de estos corales a lo largo de Chile y de la Patagonia, sea chilena que argentina, porque es una zona de corriente muy interesante. Así que estos corales van directamente, se quedarán una parte en Chile, otra parte una pequeña parte irá a la doctora Erinista.
Desde el chat nos consulta Gonzalo Bustos, ¿cuál es el programa? Hola Gonzalo. Sí, tenemos un programa, una planificación. Pretendemos, luego de estar acá en el Quisco, continuar con el cañón de San Antonio un par de días, para luego seguir tránsito hacia el sur, a la zona de Concepción, donde también hay un ecosistema muy similar acá al del Quisco, que esperamos explorar, mapear y obtener muestras. También estudiaremos el cañón del río Biobío y finalmente seguiremos hacia Isla Mocha, donde también queremos ver de qué forma la filtración que se encuentra en la zona costera podría haberse representado en ambientes más profundos. Estaremos varios días entre lo que es el cañón del Biobío, la filtración de Concepción y la Isla Mocha, esperando poder terminar recalando en el puerto de Puerto Montt el día 4 de noviembre. Es posible ver todos los días las transmisiones a través de el link del Schmidt Ocean Institute, que dice Live from the Falcor 2. Entonces, bajo la plataforma de streaming o por YouTube es posible ver la transmisión día a día de lo que estamos haciendo en el fondo del mar chileno.
Listo? Listo?
Nisky.
Listo? Can we?
As soon as we're done with this, yeah. You want to Niska? Just one or?
Just one.
Okay, sure.
Please.
Yeah. Going for a Niska. Okay.
Yeah.
Niska 4.
Zoom. Possible to put the laser lights to
They're on. Yeah, we'll see. Is that a good spot for you?
yeah, the green spot just
Yeah.
during the
Sure. Okay.
Thank you.
Okay, no problem. Okay. You want to go to waypoint 13? We're at 12 now. Okay. Okay.
Hello, on the bridge, update your waypoint.
Update, thank you.
Hey, on the bridge, we can come to, we'll stop there, we're doing some, looking at some map.
Ciao, stop.
Do you want to see this map first, or you want me to, I'll move, I have to move forward. Do you want to see the map first and then move forward? or
Okay.
just move? Okay.
Yeah. Saludamos nuevamente a los que nos están viendo y escuchando. Nos encontramos a bordo del barco oceanográfico Falcon 2 del Schmidt Ocean Institute realizando un crucero de investigación entre la desembocadura del río Limarí y Puerto Montt. Este crucero comenzó el viernes 11 de octubre zarpando desde Valparaíso y tiene por objetivo principal estudiar los ecosistemas de filtraciones de metano, cañones submarinos y chimeneas hidrotermales. El equipo científico está formado por investigadores de la Universidad de Valparaíso, de la Universidad de Boston, de la Universidad de Minnesota, de la Universidad del Estado de Oregón, de la Universidad Católica de Valparaíso. También hay científicos de la Universidad de Chile, de la Universidad Nord de Noruega y de otras instituciones interesadas en con especial afecto a uno de los principales investigadores que generaron esta propuesta que finalmente se materializó en este crucero, que es el doctor Jeffrey Marlowe de la Universidad de Boston, que no se encuentra con nosotros porque hoy día ha sido papá. Fue felizmente padre de un segundo hijo y se encuentra en Boston junto a su esposa y está cuidando a su bebé. Así que le damos un muy fuerte abrazo, un gran saludo y le deseamos que esté todo muy bien. Pero están organizando sus trabajos, sus estudiantes, que siguen principalmente la línea de lo que es la microbiología marina. Su equipo está a cargo de Erin Freitz, de Amanda Semler y de Peter Scruttle.
That's it. That's all I got.
remember once when we came up.
I know.
So I hope it's not quite like it around there.
There's supposed to be really, really big, spectacular mound at the end of these waypoints. But we have to make sure we're up for the BBC, so.
Which waypoint?
Well, they weren't sure when they told me, where are you? Are you at one of them?
We are between 13 and
13, you're here? Between 13 and 12?
Yes.
Yes.
Yeah.
Third of the way.
Where is the?
Did Jeff tell you where the really big mound was?
Nobody told me
Okay, I'm
anything.
gonna go hunt for the maps, because they didn't give me which waypoint, they just said it was over at this end.
Ah, okay.
You don't see anything really big on the sonar.
Not yet, no.
Okay. But I was just hoping you would have time to get there, because
Where is that?
huge.
Which
I don't know.
Ah.
I'm gonna find them.
Oh.
There's different atoms today, I think. Have you seen their 3D, or do you have access to the 3D map?
Can we collect the
It's gonna have to be suction.
no, well, all, but
We have
not
number
just,
5.
not just the polykit, all the structures, like,
In a bio box, yeah. There's no more quivers open.
we can put in the here.
Okay. Wants to grab that whole cluster of stuff.
Yeah.
Bio box 3. Okay.
BA, yeah.
Hopefully, I don't think it's too close for you, but we'll see.
Can I ask you to look at Jeff's screen with me to see if we can figure out? He's got the map.
Just that whole area right
Yeah.
there? Okay. Just where you're at, and Scott. I'm just gonna zoom in there. Best I can.
I suggest to from the,
Yeah, right there. So you have to scoop down a little bit more.
Am I hitting something on the board?
No. That's all slipping out of there.
Come back and get the rest.
Okay, I'll have to get the torch out.
GA.
Okay, 3A, get in there. any more of that? You just
No.
want that rock now?
Just the rock.
Okay, I get it. No more of the stuff, just the rock. You might be able to reach it with the port, actually.
Prefer,
Yeah,
for rock.
right.
Can I see your map? No. I think that's the only map that's up here right now. It doesn't show
Take the
much
piece.
of anything.
You want just the piece?
He said that,
Well, one piece first. So part later.
This is gonna be challenging to get it in that rock box. Oh, of course.
Wait, oh, not rock box.
I'm sorry, not the rock box, no, that's going in the bio box, so.
Keeping the resolver away.
Yeah,
What do you,
this is gonna be challenging, because you have to go across.
there's
the
no
top.
more boxes.
This one. It's available.
What?
So, let's see.
This one is available.
No, it's got a rock, right?
I already.
No, that's your rock. Your rock?
Yeah.
That's rubbing.
Oh, yeah, it's rubbing.
Yeah,
You're not gonna get much
BB3B.
further than that. It's gonna hit. We have to drop it in this side.
Okay.
Okay. Just put it where you can put it, Scott. Don't go down too much.
But if it falls in the other side, it's gonna mess up the other rock. I don't know what's in
There is
there.
just a tube here.
What is it? Just Unice?
Yeah, looks like Unice.
Oh, okay.
Unice there. Yeah. Oh, it's attached to the.
Fall off anyway.
What's on the rock?
There you go.
Good? Anyway, we're trying to figure out where the
Okay,
big
Scott.
mound is. I
I think
don't know
the
which
resolver's clear, I'm gonna
waypoint,
port you
but
back
you
in.
might wanna visit
Okay.
multiple waypoints. Yeah. And the time left, because they have to leave at 5 o'clock, right? For the
No.
BBC.
Okay. All right, Scott, going out on the porch. Okay. Open the file box. Yeah. Oh, there you go.
Maybe three. That's maybe.
Don't say. You follow to the waypoint. Please.
Have you been to 12 already?
We were.
And there was nothing
We are going to the.
And the 13.
That's what they just pointed to.